Hello and welcome. Do you want to successfully complete the D313 OA exam? Then you are in the right place. We built a free question bank for the D313 OA exam. Preparing for an OA is no joke, so our guides and practice questions are completely free. In this video, we will cover questions from digestive physiology, metabolic pathways, cardiorespiratory dynamics, renal function, reproductive biology. So, let's get started.
1. A pathologist examines a stomach biopsy and finds simple columnar epithelium alongside mucus-secreting cells in the tissue sample. Considering the standard layered organization of the alimentary canal, which specific wall layer was most likely captured in this biopsy specimen? A. Submucosa. B. Muscularis externa. C. Mucosa. D. Serosa. What do you think is the answer? Correct answer, C. Mucosa. Explanation. Option C is correct because the mucosa is the innermost layer of the alimentary canal, composed of simple columnar epithelium in the stomach, and is directly responsible for secreting mucus and digestive substances. Option A is incorrect because the submucosa consists of loose areolar connective tissue containing blood vessels and nerves, not the epithelial tissue with mucus-secreting cells described in this biopsy report. Option B is incorrect because the muscularis externa is composed of smooth muscle layers responsible for peristalsis and mechanical churning movements, and it would not contain epithelial or mucus-secreting cells. Option D is incorrect because the serosa is the outermost layer that functions as a smooth protective membrane, reducing friction between organs, and it does not contain the mucus-secreting epithelium found here. Let's get right into the anatomy of the alimentary canal wall. The question describes a biopsy from the stomach showing simple columnar epithelium and mucus-secreting cells. That's a dead giveaway for the mucosa, the innermost layer of the GI tract wall. The mucosa is the layer that directly faces the lumen, meaning it touches the food as it passes through. In the stomach, small intestine, and large intestine, this layer is made of simple columnar epithelium. At the two ends of the canal, the mouth and the anus, it switches to stratified squamous epithelium because those areas take more of a beating from abrasion. The mucosa also contains mucus-secreting cells, which is exactly what the biopsy found. Now, the submucosa sits just outside the mucosa and is made of loose areolar connective tissue with blood vessels. No epithelium there. The muscularis externa is all smooth muscle for peristalsis, and the serosa is the thin outermost wrapping. None of those would show simple columnar epithelium and mucus-secreting cells under a microscope. Here's a WGU D313 OA exam tip for you. When a question mentions epithelium, mucus secretion, or anything lining the lumen, the answer is always the mucosa. Quick recall hook. Mucosa equals mucus, the M's match. If it secretes mucus and faces the inside, it's the mucosa. 2. A surgeon notes that a specific layer in the stomach wall appears significantly thicker compared to the same layer found in other regions of the alimentary canal. This added thickness directly supports one of the stomach's primary mechanical roles. Which layer is most likely thicker? A. Muscularis externa. B. Mucosa. C. Submucosa. D. Serosa. Do you know the answer? Correct answer, A. Muscularis externa. Explanation. Option A is correct because the muscularis externa is notably thicker in the stomach than elsewhere in the alimentary canal, providing the powerful churning force needed for mechanical digestion of food. Option B is incorrect because while the mucosa is important for secretion and absorption throughout the canal, its thickness does not vary dramatically in the stomach to support the churning function described here. Option C is incorrect because the submucosa provides elasticity and houses blood vessels, but does not contribute to the mechanical churning movements that require the extramuscular thickness in the stomach wall. Option D is incorrect because the serosa is a thin outer membrane that reduces friction between abdominal organs and does not become significantly thicker in the stomach compared to other alimentary canal regions. Alright, this question is testing whether you know which layer of the stomach wall is beefed up compared to everywhere else in the GI tract. The answer is the muscularis externa. Here's why. The stomach doesn't just hold food, it churns it. That powerful mixing action requires thicker, smooth muscle than what you'd find in, say, the esophagus or the intestines. The muscularis externa is the layer responsible for peristalsis and segmentation throughout the entire alimentary canal. But in the stomach, it's significantly thicker because the stomach has to physically pulverize food and mix it with gastric juices to create chyme. The mucosa handles secretion and absorption, not mechanical force. The submucosa is connective tissue that provides elasticity and houses blood vessels. The serosa is just a thin, slippery coating on the outside. None of those need to be thicker in the stomach to support churning. Here's a WGU D313 OA exam tip for you. If the question talks about mechanical digestion, churning, or extra thickness in the stomach wall, think muscularis externa. Quick recall hook. Muscle equals movement. The stomach needs more muscle because it moves food harder than any other part of the canal. 3. A medical student is comparing tissue samples from the esophagus and the small intestine. She notices a key difference in the epithelial type present in the innermost wall layer of each organ. Which statement best describes this epithelial difference between these two locations? A. Both have simple columnar epithelium. B. Both have stratified squamous epithelium. C. The esophagus has simple columnar epithelium. D. The esophagus has stratified squamous epithelium. Can you guess the answer? Correct answer, D. The esophagus has stratified squamous epithelium. Explanation: Option D is correct because the esophagus is lined with stratified squamous epithelium for protection against abrasion, while the small intestine uses simple columnar epithelium suited for absorption and secretion. Option A is incorrect because simple columnar epithelium is found in the stomach and intestines for absorption and secretion, but the esophagus requires a tougher epithelial type to withstand mechanical abrasion from food. Option B is incorrect because stratified squamous epithelium is found at the ends of the alimentary canal, such as the mouth and esophagus, but the small intestine requires simple columnar for nutrient absorption. Option C is incorrect because the esophagus does not have simple columnar epithelium. It is lined with stratified squamous epithelium to protect against the friction and abrasion of food boluses passing through. This question is about a difference you need to have cold, the epithelial types at different points along the alimentary canal. The esophagus is lined with stratified squamous epithelium. The small intestine is lined with simple columnar epithelium. Two different tissues, two different jobs. Stratified squamous epithelium has multiple layers of flat cells stacked on top of each other. It's built for protection against abrasion, food scraping along as it passes through. You find it at the rough and tumble ends of the canal, the mouth, pharynx, esophagus and anus. Simple columnar epithelium, on the other hand, is a single layer of tall column-shaped cells. That thin single layer is designed for absorption and secretion, which is exactly what the stomach and intestines need to do. So when the question asks what's different between the esophagus and the small intestine, the answer is that the esophagus has stratified squamous and the small intestine has simple columnar. Here's a WGU D313 OA exam tip for you. Stratified squamous shows up at the ends of the canal where food rubs against tissue. Simple columnar shows up in the middle where nutrients need to pass through. Quick recall hook. Squamous equals scraping. Columnar equals crossing. The esophagus scrapes. The intestines absorb. 4. During an abdominal surgery, the surgeon carefully handles a membrane that both anchors the intestines to the abdominal wall and produces a lubricating fluid that prevents organ friction during digestion. Which anatomical structure is the surgeon most likely working around during this delicate procedure? A. Muscularis externa. B. Peritoneum. C. Submucosa. D. Mucosa. Do you know the answer? Correct answer, B. Peritoneum. Explanation. Option B is correct because the peritoneum is a serous membrane that anchors the intestines to the abdominal wall through mesenteries and secretes serous fluid to prevent friction between abdominal organs. Option A is incorrect because the muscularis externa is a smooth muscle layer within the wall of the alimentary canal, responsible for peristalsis, not for anchoring organs or producing lubricating serous fluid. Option C is incorrect because the submucosa is an internal layer of the alimentary canal wall containing blood vessels and connective tissue, and it does not anchor organs or secrete friction-reducing fluid. Option D is incorrect because the mucosa is the innermost lining of the alimentary canal responsible for secretion and absorption, and it plays no role in anchoring organs to the abdominal wall. This question describes a membrane that does two things. It anchors the intestines to the abdominal wall, and it secretes lubricating fluid to prevent friction between organs. That's the peritoneum. The peritoneum is a serous membrane that lines the abdominal cavity. It has two jobs you need to remember. First, it creates mesenteries, fold-like extensions that physically tether the intestines to the posterior abdominal wall so they don't just float around loose. Second, it secretes serous fluid into the peritoneal cavity. That fluid acts as a lubricant, letting organs slide past each other during digestion without generating damaging friction. The muscularis externa is a smooth muscle layer inside the wall of the GI tract. It drives peristalsis but doesn't anchor anything. The submucosa is connective tissue inside the wall that holds blood vessels. The mucosa is the innermost lining that handles secretion and absorption. None of these have anything to do with anchoring organs or producing serous fluid. Here's a WGU D313 OA exam tip for you. If the question mentions anchoring, mesenteries, serous fluid, or reducing friction between abdominal organs, the answer is peritoneum. Quick recall hook. Peritoneum equals protective wrap. It wraps, anchors, and lubricates. 5. A nursing student is studying the sequence of wall layers in the alimentary canal from the inside surface outward toward the abdominal cavity. She needs to identify the correct order for an upcoming practical examination. Which sequence correctly lists the layers from innermost to outermost? A. Mucosa submucosa, muscularis serosa. B. Submucosa mucosa muscularis serosa. C. Serosa muscularis submucosa mucosa. D. Mucosa muscularis submucosa serosa. Can you guess the answer? Correct answer. A. Mucosa submucosa muscularis serosa. Explanation. Option A is correct because the alimentary canal wall layers proceed from innermost to outermost as mucosa, submucosa, muscularis externa, and serosa, which is the standard anatomical layering sequence. Option B is incorrect because placing the submucosa before the mucosa reverses the first two layers. The mucosa is always the innermost layer that directly contacts the contents of the lumen. Option C is incorrect because this sequence lists the layers from outermost to innermost rather than from innermost to outermost, which is the exact reverse of the correct anatomical order. Option D is incorrect because this sequence swaps the positions of the muscularis and submucosa. The submucosa sits between the mucosa and the muscularis, not outside the muscularis layer. This is a straight memorization question. The order of the wall layers in the alimentary canal, from inside to outside. The correct sequence is mucosa, submucosa, muscularis, serosa. Start from the lumen and work your way out. The mucosa is first because it directly lines the interior. Right outside the mucosa is the submucosa, which provides support with its connective tissue and blood vessels. Next comes the muscularis externa, the smooth muscle layer that generates peristalsis and churning. The outermost layer is the serosa, a thin serous membrane that forms the external surface and reduces friction. A common mistake is swapping the submucosa and the muscularis. Remember that the submucosa always sits between the mucosa and the muscularis. It never jumps outside the muscle layer. Here's a WGU D313 OA exam tip for you. Memorize the order as a phrase: M sub muscsa, mucosa, submucosa, muscularis, serosa, inside to out. Quick recall hook. Think my stomach makes sounds. Mucosa, submucosa, muscularis, serosa. First letter of each word matches. 6. A researcher is studying the cellular mechanisms that create the highly acidic environment inside the stomach needed for protein digestion. She identifies the specific cell type responsible for secreting both hydrochloric acid and a glycoprotein essential for vitamin absorption. Which gastric cell type is she studying? A. Chief cells, B. G cells, C. Surface mucous cells, D. Parietal cells. Can you guess the answer? Correct answer: D. Parietal cells. Explanation: option D is correct because parietal cells secrete hydrochloric acid to create the acidic stomach environment and also produce intrinsic factor, the glycoprotein required for vitamin B12 absorption in the ileum. Option A is incorrect because chief cells secrete pepsinogen, the inactive precursor enzyme that must be activated by hydrochloric acid to become pepsin, but they do not produce acid or intrinsic factor. Option B is incorrect because G cells are enteroendocrine cells that secrete the hormone gastrin, which stimulates other cells to release acid and enzymes, but G cells do not directly produce hydrochloric acid. Option C is incorrect because surface mucous cells secrete mucin to protect the stomach lining from its own acidic contents, but they are not responsible for producing hydrochloric acid or intrinsic factor. This question gives you 2 clues about a single cell type. It secretes hydrochloric acid and it produces a glycoprotein needed for vitamin absorption. Both of those functions belong to parietal cells. Parietal cells sit in the gastric glands of the stomach mucosa. Their first job is pumping out HCl, which drops the stomach pH to around 1.5 to 2. That acidic environment is needed to activate pepsinogen into pepsin so protein digestion can actually begin. Their second job is producing intrinsic factor, a glycoprotein that latches onto vitamin B12 and escorts it all the way to the ileum, where B12 gets absorbed. Without intrinsic factor, you develop pernicious anemia because B12 can't be taken up. Chief cells are the wrong pick because they make pepsinogen, not acid or intrinsic factor. G cells secrete gastrin, a hormone that tells other cells to produce acid. They don't produce acid themselves. Surface mucous cells make mucin to protect the stomach lining. Here's a WGU D313 OA exam tip for you. When a question mentions HCl, intrinsic factor, or vitamin B12 absorption, point straight to parietal cells. Quick recall hook: parietal equals pair. They do a pair of things: acid and intrinsic factor. 7. A patient develops a condition where the cells responsible for producing the inactive enzyme precursor in the stomach are destroyed by an autoimmune disease. Without these cells functioning, the initial breakdown of dietary protein in the stomach would be most directly impaired. Which cell type has been destroyed? A. Parietal cells, B. G cells, C. Chief cells, D. Enteroendocrine cells. Can you guess the answer? Correct answer: C. Chief cells. Explanation: option C is correct because chief cells produce pepsinogen, the inactive enzyme precursor that is converted to pepsin by hydrochloric acid, making them essential for initiating protein digestion within the stomach. Option A is incorrect because parietal cells produce hydrochloric acid and intrinsic factor, While acid loss would also impair digestion, the question specifically asks about the cells producing the inactive enzyme precursor. Option B is incorrect because G cells secrete the hormone, gastrin, that stimulates acid and enzyme production, but they do not themselves produce the inactive enzyme precursor pepsinogen described in this scenario. Option D is incorrect because enteroendocrine cells secrete regulatory hormones such as serotonin, histamine, and somatostatin that modulate stomach activity, but they do not produce pepsinogen or any digestive enzyme precursors. This scenario is about autoimmune destruction of the cells that make the inactive enzyme precursor in the stomach. That precursor is pepsinogen, and the cells that produce it are chief cells. Here's the chain of events in a healthy stomach. Chief cells release pepsinogen into the stomach lumen. Parietal cells release hydrochloric acid. The acid hits pepsinogen and converts it into pepsin, which is the active form that starts chopping up proteins. If you wipe out chief cells, you lose the pepsinogen supply entirely. No pepsinogen means no pepsin. No pepsin means protein digestion in the stomach is dead in the water. Parietal cells make HCl and intrinsic factor. Losing them would also be a disaster, but the question specifically asks about the inactive enzyme precursor, and that's pepsinogen, not acid. G cells make gastrin, a hormone. Enteroendocrine cells make regulatory hormones like serotonin and histamine. Neither of those are enzyme precursors. Here's a WGU D313 OA exam tip for you. The phrase inactive enzyme precursor is code for pepsinogen, and pepsinogen always points to chief cells. Quick recall hook. Chief starts with CH. So does chemical precursor. Chief cells are the precursor factory. 8. When food first enters the stomach and triggers distension, a specific hormone is released that creates a positive feedback loop by stimulating both acid secretion and further enzyme production. This hormone also promotes stomach wall contractions. Which hormone initiates this cascade of gastric activity? A. Secretin. B. Gastrin. C. Somatostatin. D. Cholecystokinin. Can you guess the answer? Correct answer, B. Gastrin. Explanation: option B is correct because gastrin is released by G cells when food enters the stomach, and it stimulates parietal cells to release hydrochloric acid, chief cells to release pepsinogen, and promotes stomach contractions. Option A is incorrect because secretin is released by the duodenum in response to acidic chyme and stimulates the pancreas to secrete bicarbonate rather than driving acid production inside the stomach itself. Option C is incorrect because somatostatin acts to slow down stomach activity rather than stimulate it, functioning as an inhibitory hormone that counterbalances the stimulatory effects of gastrin on gastric secretions. Option D is incorrect because cholecystokinin is released from the duodenum and primarily stimulates gallbladder contraction and pancreatic enzyme secretion, not the initial cascade of acid and enzyme production in the stomach. The question is describing a hormone that kicks off a positive feedback loop when food hits the stomach. That hormone is gastrin. When food enters the stomach and stretches the walls, G cells in the stomach mucosa release gastrin into the blood. Gastrin then does three things at once. It tells parietal cells to release hydrochloric acid, it tells chief cells to release pepsinogen, and it stimulates contractions of the stomach wall. More acid means more pepsinogen gets activated into pepsin, which means more protein breakdown, which keeps the whole cycle rolling. That's the positive feedback loop the question is describing. Secretin isn't the right choice because it's released from the duodenum and makes the pancreas secrete bicarbonate. It's a downstream hormone, not a stomach starter. Somatostatin actually slows stomach activity. It's the brake, not the gas pedal. CCK is also released from the duodenum and triggers gallbladder contraction and pancreatic enzyme release, not gastric acid production. Here's a WGU D313 OA exam tip for you. When a question mentions food entering the stomach, positive feedback, acid production, and stomach contractions all happening together, The answer is gastrin. Quick recall hook. Gastrin starts the gastric party. Same route, same location, same job. 9. The stomach produces extremely strong acid that could damage its own tissue if left unprotected. A specific cell type in the gastric glands secretes a protective substance that coats the stomach lining and prevents self-digestion by this corrosive environment. Which cell type provides this essential protection? A. Surface mucous cells. B. Chief cells. C. Parietal cells. D. G cells. Do you know the answer? Correct answer, A. Surface mucous cells. Explanation. Option A is correct because surface mucous cells secrete mucin, which forms a protective mucous barrier over the stomach lining, shielding the underlying mucosa from the corrosive effects of hydrochloric acid. Option B is incorrect because chief cells are responsible for secreting pepsinogen, the inactive form of the protein-digesting enzyme pepsin, and they do not produce the protective mucus that shields the stomach wall. Option C is incorrect because parietal cells are actually the source of the hydrochloric acid that creates the corrosive environment, making them the cause of the threat rather than the provider of protection. Option D is incorrect because G cells secrete the hormone gastrin that stimulates further acid production and stomach contractions, which would increase rather than protect against the corrosive gastric environment. The stomach produces hydrochloric acid strong enough to dissolve metal, so it obviously needs a defense system to keep from digesting itself. That defense comes from surface mucous cells. These cells sit in the gastric glands and secrete mucin, which forms a thick, sticky mucus barrier that coats the entire inner surface of the stomach. This gel-like layer physically separates the corrosive acid and pepsin from the delicate mucosal tissue underneath. If this mucus layer breaks down, which is what happens with H. pylori infections or overuse of NSAIDs, the acid eats into the stomach wall and you get ulcers. Chief cells make pepsinogen, which actually contributes to the digestive threat rather than protecting against it. Parietal cells are the source of the acid itself. G cells release gastrin, which stimulates even more acid production. None of those are protective. Here's a WGU D313 OA exam tip for you. Any question about protecting the stomach lining from acid damage, preventing self-digestion, or a mucus barrier is pointing at surface mucous cells. Quick recall hook. Mucous cells make mucus shield. They're the stomach's bodyguard. 10. A clinician is explaining how pepsinogen activation works to a medical student. She describes a cascade where one cell type creates the chemical conditions that transform a product from another cell type into its active form. Which statement best describes this activation cascade in the stomach? A. Gastrin directly converts pepsinogen to pepsin. B. Chief cells secrete acid that activates pepsin. C. Mucous cells activate pepsinogen with mucin. D. Hydrochloric acid converts pepsinogen to pepsin. Can you guess the answer? Correct answer. D. Hydrochloric acid converts pepsinogen to pepsin. Explanation. Option D is correct because hydrochloric acid, secreted by parietal cells, converts the inactive pepsinogen released by chief cells into pepsin, the active protein digesting enzyme that initiates protein breakdown in the stomach. Option A is incorrect because gastrin is a hormone secreted by G cells that stimulates acid and enzyme production, but does not directly convert pepsinogen into its active form pepsin through chemical interaction. Option B is incorrect because chief cells secrete pepsinogen, not hydrochloric acid. It is the parietal cells that produce the acid responsible for activating pepsinogen into the active enzyme pepsin. Option C is incorrect because mucous cells secrete mucin to protect the stomach lining from acid damage, and mucin plays no role in the chemical conversion of pepsinogen into its active enzyme form. This question is testing whether you understand the pepsinogen activation cascade specifically that hydrochloric acid converts pepsinogen into pepsin. Two cell types are involved and they work as a team. Chief cells release pepsinogen, which is an inactive enzyme. On its own, pepsinogen can't digest anything. It needs to be activated. That activation happens when parietal cells pump hydrochloric acid into the stomach lumen. The acid environment cleaves pepsinogen into pepsin, the active protein digesting enzyme. This is a classic biochemical cascade. One cell makes the substrate, another cell creates the chemical conditions to activate it. Gastrin doesn't directly convert pepsinogen. It's a hormone that tells the cells to start working. Chief cells don't make acid. They make the precursor. Mucous cells have nothing to do with enzyme activation. Here's a WGU D313 OA exam tip for you. The activation cascade in the stomach always follows this chain. Chief cells release pepsinogen, parietal cells release HCl. HCl converts pepsinogen to pepsin. If the question asks what activates pepsinogen, the answer is always hydrochloric acid. Quick recall hook: HCl is the key that unlocks pepsin. No acid, no activation. 11. A physiology professor explains that the small intestine achieves an enormous absorptive surface area of roughly 200 square meters through three specific structural modifications working together. A student asks which adaptation consists of tiny hair-like extensions on epithelial cell surfaces, collectively called the brush border. Which structure is the professor describing? A. Circular folds. B. Villi. C. Microvilli. D. Rugae. Can you guess the answer? Correct answer, C. Microvilli. Explanation: option C is correct because microvilli are the tiny hair-like extensions on the surface of each epithelial cell lining the villi, and they are collectively referred to as the brush border of the small intestine. Option A is incorrect because circular folds are permanent macroscopic ridges in the intestinal wall that increase surface area at a larger structural level, but they are not the hair-like cellular extensions called brush border. Option B is incorrect because villi are finger-like projections of the mucosa that give the interior a velvety appearance, but they are larger structures than microvilli and are not themselves called the brush border. Option D is incorrect because rugae are the folds found on the interior surface of the stomach that allow expansion when filled with food, and they are not a structural feature of the small intestine. The question asks which structural adaptation of the small intestine consists of tiny hair-like extensions on epithelial cell surfaces called the brush border. That's microvilli. The small intestine has three layers of surface area amplification stacked on top of each other. First, there are circular folds. These are large, permanent ridges in the intestinal wall that you can see without a microscope. Second, there are villi, finger-like projections of the mucosa that stick up from those folds, giving the intestinal lining a velvety look to the naked eye. Third, there are microvilli, microscopic hair-like extensions on the surface of each individual epithelial cell sitting on top of each villus. When you look at all those microvilli packed together, they form what's called the brush border, because under a microscope it looks like the bristles of a tiny brush. Together, these three adaptations expand the absorptive surface to roughly 200 square meters. Rugae are folds in the stomach, not the small intestine, so that option is a trap. Here's a WGU D313 OA exam tip for you. Brush border is always microvilli. Any question that uses that phrase is pointing to the smallest of the three surface area adaptations. Quick recall hook: micro means tiny. Microvilli are the tiniest projections, and they form the brush border. 12. A patient with a vitamin deficiency is diagnosed with impaired absorption of vitamin B12. The gastroenterologist explains that this specific vitamin requires intrinsic factor and is absorbed in one particular subdivision of the small intestine. Which subdivision is responsible for absorbing this critical vitamin with the help of intrinsic factor? A. Ileum. B. Duodenum. C. Jejunum. D. Cecum. Can you guess the answer? Correct answer, A. Ileum. Explanation. Option A is correct because the ileum is the last subdivision of the small intestine and is specifically responsible for absorbing vitamin B12, which requires the binding of intrinsic factor produced by parietal cells. Option B is incorrect because the duodenum is the first and shortest subdivision of the small intestine where most chemical digestion takes place, but it is not the primary site for vitamin B12 absorption. Option C is incorrect because the jejunum is the middle subdivision of the small intestine where most general nutrient absorption occurs, but the specific absorption of vitamin B12 with intrinsic factor happens in the ileum. Option D is incorrect because the cecum is the first part of the large intestine that receives material from the ileum through the ileocecal valve, and it is not a subdivision of the small intestine at all. The question asks which part of the small intestine absorbs vitamin B12 with the help of intrinsic factor. The answer is the ileum. The small intestine has three subdivisions in order: duodenum, jejunum, and ileum. Each one has a specialty. The duodenum is the first and shortest section. It's where the heavy-duty chemical digestion happens, because that's where bile and pancreatic juices get dumped in. The jejunum is the middle section and handles most general nutrient absorption. The ileum is the longest section and the final stretch before the large intestine. Its special job is absorbing vitamin B12, and it can only do this when B12 is bound to intrinsic factor, a glycoprotein produced by parietal cells back in the stomach. Without intrinsic factor, B12 passes right through the ileum, unabsorbed, leading to pernicious anemia. The cecum is a trick answer because it's the first part of the large intestine, not a subdivision of the small intestine at all. Here's a WGU D313 OA exam tip for you. B12 and intrinsic factor always go with the ileum. If you see either term, pick ileum. Quick recall hook: ileum absorbs the important vitamin. I for ileum, I for intrinsic factor. 13. During a lecture on nutrient absorption, a professor explains that most digested nutrients enter the capillaries within the villi and travel directly into the bloodstream. However, one major nutrient class takes a completely different absorption route through specialized lymphatic vessels. Which nutrient class follows this alternative lymphatic pathway? A. Carbohydrates, B. Lipids, C. Proteins, D. Minerals. Can you guess the answer? Correct answer, B. Lipids. Explanation. Option B is correct because lipids are absorbed into lacteals, which are specialized lymphatic vessels within the intestinal villi, and they reach the systemic blood circulation through the lymphatic system rather than directly. Option A is incorrect because carbohydrates are broken down into monosaccharides that are absorbed directly into the capillaries of the villi and transported through the bloodstream to cells throughout the body. Option C is incorrect because proteins are broken down into individual amino acids that cross into the capillaries of the villi and enter the bloodstream directly, following the same pathway as most other absorbed nutrients. Option D is incorrect because minerals are absorbed in the small intestine and pass into the blood capillaries of the villi for transport, not into the lymphatic lacteals that are specifically used for lipid absorption. Most digested nutrients, carbohydrates broken into monosaccharides, proteins broken into amino acids, and minerals, all get absorbed into the capillaries inside the villi and go directly into the bloodstream. Lipids are the exception. After fats are digested by pancreatic lipase into fatty acids and monoglycerides, they get absorbed into the intestinal epithelial cells, reassembled into triglycerides, packaged into chylomicrons, and then sent into the lacteals. Those are the tiny lymphatic vessels inside each villus. From there, the fats travel through the lymphatic system and eventually get dumped into the bloodstream via the thoracic duct. The reason fats take this alternate route is because they're hydrophobic. They don't dissolve well in blood plasma, so they need to be packaged into lipoprotein particles first and routed through the lymphatic system. Carbohydrates, proteins, and minerals all go straight into capillaries. No lymphatic detour needed. Or here's a WGU D313 OA exam tip for you. If a question asks about an alternative absorption pathway, lymphatic vessels or lacteals, the answer is lipids. Quick recall hook. Lipids love lacteals. L and L go together. Fats go lymphatic. 14. Highly acidic chyme entering the duodenum from the stomach could severely damage the intestinal wall if not neutralized quickly. A specific set of glands found exclusively in the duodenum secretes an alkaline mucus that counteracts this acidity and protects the tissue. Which glands perform this critical neutralizing function? A. Crypts of Lieberkühn. B. Gastric glands. C. Salivary glands. D. Brunner's glands. What do you think is the answer? Correct answer, D. Brunner's glands. Explanation: option D is correct because Brunner's glands are found exclusively in the duodenum and secrete an alkaline mucus that neutralizes the acidic chyme arriving from the stomach, thereby protecting the intestinal wall from damage. Option A is incorrect because the crypts of Lieberkühn are glands located at the base of the villi throughout the small intestine that secrete lysozyme for antimicrobial defense, not the alkaline mucus needed for neutralization. Option B is incorrect because gastric glands are located in the stomach mucosa and contain cells that produce hydrochloric acid and pepsinogen, making them a source of acidity rather than a neutralizing structure. Option C is incorrect because salivary glands are located in the oral cavity and produce saliva containing salivary amylase for starch digestion, and they play no role in neutralizing acid in the duodenum. When acidic chyme fires out of the stomach through the pyloric sphincter and hits the duodenum, it could burn right through the intestinal wall if nothing neutralized it. That's where Brunner's glands come in. Brunner's glands are found exclusively in the duodenum. You won't find them anywhere else in the GI tract. They secrete an alkaline mucus that coats the duodenal lining and counteracts the acid arriving from the stomach. This is a localized defense mechanism right at the point where damage would be worst. Crypts of Lieberkühn are glands at the base of the villi throughout the small intestine, but they secrete lysozyme for antimicrobial protection, not alkaline mucus for acid neutralization. Gastric glands are in the stomach and produce acid, so they're part of the problem, not the solution. Salivary glands are in the mouth and have nothing to do with the duodenum. Here's a WGU D313 OA exam tip for you. If the question says duodenum only and alkaline mucus, the answer is Brunner's glands every time. Quick recall hook: Brunner's buffers the burn. The acid burns and Brunner's glands buffer it. 15. A student is tracing the path that food follows as it moves from the stomach through the entire small intestine and into the large intestine. She needs to list every structure in the correct sequence for her anatomy examination. Which sequence correctly describes this complete pathway? A. Duodenum, jejunum, ileum, ileocecal valve. B. Ileocecal valve, duodenum, ileum, jejunum. C. Pyloric sphincter, duodenum, jejunum, ileum. D. Jejunum, duodenum, ileum, ileocecal valve. Can you guess the answer? Correct answer, C. Pyloric sphincter, duodenum, jejunum, ileum. Explanation: option C is correct because food exits the stomach through the pyloric sphincter, then passes sequentially through the duodenum, jejunum, and ileum, before reaching the ileocecal valve and entering the large intestine. Option A is incorrect because while the duodenum, jejunum, and ileum are in the correct internal order, this sequence omits the pyloric sphincter that controls passage from the stomach into the duodenum initially. Option B is incorrect because the ileocecal valve is located at the end of the small intestine where it connects to the large intestine, not at the beginning of the pathway from the stomach into the duodenum. Option D is incorrect because this sequence places the jejunum before the duodenum, which reverses the first two subdivisions. The duodenum is always the first section receiving chyme from the stomach. This question wants the complete pathway from the stomach through the entire small intestine and into the large intestine. The correct sequence starts at the pyloric sphincter. That's the muscular ring that controls the exit from the stomach into the first section of the small intestine. After passing through the pyloric sphincter, food enters the duodenum. From the duodenum it moves into the jejunum. From the jejunum it continues into the ileum. After the ileum it crosses the ileocecal valve and enters the cecum of the large intestine. The key here is that the answer choice includes the pyloric sphincter as the starting point, which is correct because the question says from the stomach through the entire small intestine. Option A lists duodenum, jejunum, ileum, ileocecal valve. That's the right internal order, but it misses the pyloric sphincter at the start. Option B puts the ileocecal valve first, which is completely backward. Option D puts the jejunum before the duodenum, which is wrong because the duodenum is always first. Here's a WGU D313 OA exam tip for you. The small intestine order is always DJI. Duodenum, jejunum, ileum. From shortest to longest, same order. Quick recall hook, DJI spins the track. DJI, always in that order, always starting with the pyloric sphincter. 16. A dietitian explains to a patient that a specific substance produced by the body's largest gland does not contain digestive enzymes, but instead breaks large fat globules into smaller droplets, dramatically increasing the surface area available for enzymatic fat digestion. Which substance is the dietitian describing, and what organ produces it? A. Bile produced by the liver. B. Pepsinogen produced by the stomach. C. Bicarbonate produced by the pancreas. D. Lipase produced by the pancreas. Do you know the answer? Correct answer, A. Bile produced by the liver. Explanation: Option A is correct, because bile is produced by the liver, the body's largest gland, and it emulsifies fats by breaking large globules into smaller droplets without containing any digestive enzymes itself. Option B is incorrect because pepsinogen is an inactive enzyme precursor produced by chief cells in the stomach that is converted to pepsin for protein digestion, and it has no role in fat emulsification. Option C is incorrect because bicarbonate ions are produced by the pancreas to neutralize acidic chyme entering the duodenum, but bicarbonate does not break fat globules into smaller droplets for enzymatic digestion. Option D is incorrect because while pancreatic lipase does digest fats, it is itself a digestive enzyme that breaks down triglycerides. The question specifically describes a substance that contains no enzymes and works through emulsification. This question describes a substance from the body's largest gland that contains no digestive enzymes but breaks fat globules into smaller droplets. That substance is bile, and the organ is the liver. The liver is the largest gland in the human body, located just below the diaphragm on the right side. One of its many jobs is producing bile, a yellow-green fluid that gets stored in the gallbladder until it's needed. When fatty food hits the duodenum, CCK triggers the gallbladder to contract and squirt bile into the duodenum. Bile then emulsifies fats. It breaks large fat globules into many tiny droplets. This matters because pancreatic lipase can only work on the surface of fat droplets, so the more surface area you create, the faster lipase can break down the fat. The critical detail? Bile is not a digestive enzyme. It doesn't break chemical bonds. It physically disperses fat globules like dish soap breaking up grease. Pepsinogen is a stomach enzyme precursor for protein digestion. Bicarbonate neutralizes acid but doesn't touch fat. Lipase is an actual enzyme that breaks fat down chemically. Here's a WGU D313 OA exam tip for you. No enzymes plus fat emulsification plus largest gland is a triple lock on bile from the liver. Quick recall hook, bile breaks fat into bits. 3 Bs, emulsification, not digestion. 17. A patient is diagnosed with a complete blockage at the hepatopancreatic ampulla, preventing any secretions from reaching the duodenum through this shared opening. The gastroenterologist explains that this blockage will impair 2 critical digestive functions simultaneously. Which pair of functions would be most directly compromised? A. Protein digestion and water absorption. B. Starch digestion and vitamin absorption. C. Mucus secretion and hormone release. D. Fat emulsification and chyme neutralization. What do you think is the answer? Correct answer. D. Fat emulsification and chyme neutralization. Explanation. Option D is correct because a blockage at the hepatopancreatic ampulla prevents both bile from reaching the duodenum for fat emulsification and pancreatic bicarbonate from neutralizing the acidic chyme entering from the stomach. Option A is incorrect because protein digestion begins in the stomach with pepsin and continues with pancreatic enzymes, while water absorption primarily occurs in the large intestine, neither being the pair most directly blocked. Option B is incorrect because while pancreatic amylase for starch digestion would be affected, vitamin absorption occurs throughout the small intestine independently and is not directly impaired by a blockage at the hepatopancreatic ampulla. Option C is incorrect because mucus secretion from goblet cells and Brunner's glands occurs locally within the intestinal wall, and hormonal release from enteroendocrine cells is also independent of the hepatopancreatic ampulla. A blockage at the hepatopancreatic ampulla is a 2-for-1 disaster because both bile and pancreatic juice share this opening into the duodenum. If nothing can get through, you lose 2 functions simultaneously. First, bile can't reach the duodenum, so fat emulsification stops. Without bile breaking fat globules into tiny droplets, pancreatic lipase has almost nothing to work with. Second, pancreatic juice containing bicarbonate can't reach the duodenum, so the acidic chyme from the stomach goes unneutralized. That acid can damage the intestinal wall. So the pair of functions lost is fat emulsification and chyme neutralization. Protein digestion and water absorption doesn't fit because protein digestion starts in the stomach, with pepsin, and water absorption happens mainly in the large intestine. Starch digestion and vitamin absorption doesn't work either. Starch digestion would be partially affected, but vitamin absorption isn't directly tied to this ampulla. Mucus secretion and hormone release happen locally within the intestinal wall, independent of the ampulla. Here's a WGU D313 OA exam tip for you. Hepatopancreatic ampulla blockage always means 2 things are lost: bile for fat and bicarbonate for acid. Fat emulsification and chyme neutralization is the answer. Quick recall hook: hepato means liver, pancreatic means pancreas. Block both at once, lose both their products. 18. A medical student is studying the dual functionality of the pancreas. She learns that one portion secretes digestive enzymes through a duct into the duodenum, while another portion releases hormones directly into the bloodstream. Which description correctly identifies the cell type responsible for the endocrine function of regulating blood glucose during the absorptive state? A. Centroacina cells secreting bicarbonate. B. Beta cells secreting insulin. C. Alpha cells secreting glucagon. D. Axina cells secreting pancreatic amylase. Can you guess the answer? Correct answer. B. Beta cells secreting insulin. Explanation. Option B is correct because beta cells in the pancreatic islets of Langerhans perform the endocrine function of secreting insulin directly into the blood, which moves glucose into cells during the absorptive state. Option A is incorrect because centroacina cells are part of the exocrine pancreas and secrete bicarbonate ions into the pancreatic duct to neutralize acidic chyme rather than releasing hormones into the bloodstream. Option C is incorrect because alpha cells secrete glucagon, which mobilizes stored glucose into the bloodstream during the fasting or post-absorptive state, not during the absorptive state when blood glucose is already elevated. Option D is incorrect because acinar cells are exocrine cells that produce and secrete digestive enzymes into the pancreatic duct for delivery to the duodenum, and they do not release hormones into the bloodstream. The pancreas pulls double duty. It's both an exocrine and an endocrine organ. The exocrine part secretes digestive enzymes through a duct into the duodenum. The endocrine part releases hormones directly into the bloodstream. The question specifically asks about the endocrine function during the absorptive state, which is the fed state after eating when blood glucose is high. That points directly to beta cells secreting insulin. Beta cells live in the islets of Langerhans inside the pancreas. When blood glucose rises after a meal, beta cells detect it and release insulin. Insulin then moves glucose from the blood into cells for energy use or storage. That's the absorptive state in a nutshell. Insulin is dominant, glucose is being cleared from blood. Centroacinar cells secrete bicarbonate into the pancreatic duct. That's exocrine, not endocrine. Alpha cells secrete glucagon, which does the opposite of insulin. It raises blood glucose during fasting, not the absorptive state. Acinar cells produce digestive enzymes and secrete them into the duct, are also exocrine. Here's a WGU D313 OA exam tip for you. Absorptive state equals fed state equals insulin equals beta cells. If the question says after eating or absorptive, think beta cells and insulin. Quick recall hook. B for beta, B for blood sugar goes down. Beta cells lower blood glucose with insulin. 19. A surgeon is tracing the pathway that bile follows from its storage location to the duodenum during fat digestion. She needs to identify the specific duct that carries bile from the gallbladder before it merges with the common hepatic duct. Which duct transports bile specifically from the gallbladder into the shared ductal system? A. Pancreatic duct. B. Common bile duct. C. Cystic duct. D. Common hepatic duct. What do you think is the answer? Correct answer, C. Cystic duct. Explanation: option C is correct because the cystic duct is the specific channel that carries bile from the gallbladder, and it joins the common hepatic duct to form the common bile duct that delivers bile to the duodenum. Option A is incorrect because the pancreatic duct carries digestive enzymes and bicarbonate from the pancreas to the duodenum, and it is a completely separate structure from the gallbladder's own drainage duct system. Option B is incorrect because the common bile duct is the structure formed after the cystic duct merges with the common hepatic duct. It is the result of the merger rather than the duct leaving the gallbladder. Option D is incorrect because the common hepatic duct carries bile from the liver itself, not from the gallbladder, and it is the structure that the cystic duct joins to form the common bile duct downstream. The question is tracing the path bile takes from the gallbladder to the duodenum and asks you to name the duct that leaves the gallbladder specifically. That's the cystic duct. Here's how the ductal system works. The liver produces bile continuously and drains it through the right and left hepatic ducts, which merge into the common hepatic duct. The gallbladder, sitting underneath the liver, stores and concentrates that bile. When it's time to release bile during fat digestion, the gallbladder contracts and pushes bile out through the cystic duct. The cystic duct then joins the common hepatic duct to form the common bile duct, which delivers bile into the duodenum. The pancreatic duct is a separate structure from the pancreas. It carries enzymes and bicarbonate, not bile from the gallbladder. The common bile duct is the structure formed after the merger, not the duct leaving the gallbladder. The common hepatic duct carries bile from the liver, not from the gallbladder. Here's a WGU D313 OA exam tip for you. Cystic duct connects to the cyst-like gallbladder. If the question asks what duct leaves the gallbladder, cystic duct is the answer. Quick recall hook. Cystic sounds like cyst. The gallbladder looks like a cyst. Cystic duct drains the gallbladder. 20. A patient with chronic pancreatic inflammation has experienced progressive destruction of specific endocrine cells within the organ. Her physician explains that the most clinically significant consequence of this destruction involves the loss of a hormone critical for moving glucose from the blood into cells. Which condition is this patient most likely to develop? A. Type 1 diabetes from beta cell loss. B. Pernicious anemia from intrinsic factor loss. C. Scurvy from vitamin C deficiency. D. Muscle wasting from protein deficiency. Do you know the answer? Correct answer, AA, type 1 diabetes from beta cell loss. Explanation. Option A is correct because chronic pancreatic inflammation can destroy beta cells in the islets of Langerhans, eliminating insulin production and leading to type 1 diabetes where glucose cannot enter cells properly. Option B is incorrect because pernicious anemia results from a deficiency of intrinsic factor produced by parietal cells in the stomach, not from damage to any cell type within the pancreas or its endocrine tissue. Option C is incorrect because scurvy results from a dietary deficiency of vitamin C affecting collagen synthesis, and it has no connection to pancreatic inflammation or the destruction of pancreatic endocrine cells. Option D is incorrect because muscle wasting from protein deficiency results from inadequate dietary protein intake or prolonged starvation, not from the destruction of hormone-producing endocrine cells within the pancreas. Chronic pancreatic inflammation destroying endocrine cells. The question is asking what happens when you lose the cells that make insulin. Those are beta cells in the islets of Langerhans. When beta cells get destroyed, insulin production stops. Without insulin, glucose stays trapped in the bloodstream because insulin is the hormone that opens the door for glucose to enter cells. The result is type 1 diabetes. The body has plenty of glucose floating around in the blood, but cells are starving for energy because they can't take it in. This is different from type 2 diabetes, where cells become resistant to insulin that is still being produced. Here the beta cells are physically destroyed, so no insulin is made at all. Pernicious anemia comes from losing intrinsic factor, which is produced by parietal cells in the stomach, nothing to do with the pancreas. Scurvy comes from not eating enough vitamin C. Muscle wasting comes from protein deficiency during starvation. None of those are caused by pancreatic endocrine cell destruction. Here's a WGU D313 OA exam tip for you. Destroyed pancreatic endocrine cells plus lost glucose regulation equals type 1 diabetes from beta cell destruction. That's the connection to lock in. Quick recall hook. Beta cells are broken in type 1. B for beta, B for broken. No beta cells, no insulin, type 1 diabetes. 21. A physiology instructor asks her class to identify where the most extensive phase of chemical digestion occurs in the body. She explains that this location receives enzymatic secretions from an accessory organ and also contains its own membrane-bound enzymes that complete the final breakdown of all macronutrient categories. Which location is she describing? A. Stomach. B. Oral cavity. C. Large intestine. D. Small intestine. Do you know the answer? Correct answer, D. Small intestine. Explanation. Option D is correct because the small intestine is where the most extensive chemical digestion occurs, receiving pancreatic enzymes and using its own brush border enzymes to complete the breakdown of all three macronutrients. Option A is incorrect because the stomach primarily digests protein through the action of pepsin and performs heavy mechanical digestion, but it does not complete the chemical breakdown of all macronutrient categories. Option B is incorrect because the oral cavity only initiates carbohydrate digestion through salivary amylase acting on starches, and it does not receive accessory organ secretions or break down proteins or fats. Option C is incorrect because the large intestine primarily absorbs water and electrolytes and houses bacteria that ferment remaining fibers, but it does not perform significant enzymatic chemical digestion of macronutrients. This is a foundational concept you'll see tested in different ways. Where does the most extensive chemical digestion happen? The answer is the small intestine. The oral cavity only starts carbohydrate digestion with salivary amylase. The stomach handles protein digestion with pepsin and does serious mechanical work, but it doesn't finish the job on all three macronutrients. The large intestine is mainly about water absorption and bacterial fermentation of leftover fiber. The small intestine is where all three macronutrient classes, carbohydrates, proteins, and fats, get fully broken down. It receives pancreatic enzymes through the pancreatic duct, pancreatic amylase for carbs, pancreatic lipase for fats, and trypsin for proteins. On top of that, the small intestine has its own brush border enzymes embedded in the microvilli membranes that finish the breakdown into final, absorbable forms: monosaccharides, amino acids, fatty acids. No other location in the GI tract receives accessory organ secretions and has its own membrane-bound enzymes working together like this. Here's a WGU D313 OA exam tip for you. If the question says most extensive chemical digestion, or all macronutrients, the answer is the small intestine. Quick recall hook. Small intestine does the big work. Don't let the word small fool you. 22. A nursing student is studying for an exam on digestive enzymes and their specific target molecules. She needs to correctly match each enzyme category to the macronutrient it breaks down. Which pairing correctly identifies an enzyme class with its corresponding substrate in the digestive system? A. Lipases break down carbohydrates. B. Amylases break down carbohydrates. C. Proteases break down lipids. D. Amylases break down proteins. Do you know the answer? Correct answer: B. Amylases break down carbohydrates. Explanation: Option B is correct because amylases are the enzyme class that specifically targets and breaks down carbohydrates, including salivary amylase in the mouth and pancreatic amylase in the small intestine during digestion. Option A is incorrect because lipases are the enzyme class that specifically breaks down lipids or fats into fatty acids and monoglycerides, not carbohydrates, with pancreatic lipase being the primary example in digestion. Option C is incorrect because proteases are the enzyme class that breaks down proteins into smaller peptides and amino acids, not lipids, with pepsin and trypsin being the major proteases in the digestive system. Option D is incorrect because amylases specifically target carbohydrates such as starches and polysaccharides, not proteins. The enzyme class responsible for protein digestion is the proteases, including pepsin and trypsin. This question tests a simple but high-yield matching rule. Which enzyme class breaks down which macronutrient? The correct pairing is amylases break down carbohydrates. There are three enzyme classes you need to know cold. Amylases target carbohydrates. Salivary, amylase in the mouth, and pancreatic amylase in the small intestine both chop up starches and polysaccharides into smaller sugars. Lipases target fats. Pancreatic lipase is the main one, breaking triglycerides into fatty acids and monoglycerides in the small intestine. Proteases target proteins. Pepsin in the stomach and trypsin in the small intestine break peptide bonds. The naming convention is your cheat code here. Amylase and amylose. Amylose is a type of starch, so amylase breaks starch. Lipase has lip in it. Lipids, protease has protein in it, proteins. The wrong answers in this question just scramble those pairings to trip you up. Here's a WGU D313 OA exam tip for you. Enzyme names tell you what they break down. Amylase equals carbs, lipase equals fats, protease equals proteins. Quick recall hook. ALP matches CFP. Amylase, lipase, protease breaks, carbs, fats, proteins. 23. A patient takes a bite of bread and begins chewing. Her instructor explains that a specific enzyme in her saliva immediately begins breaking the chemical bonds in the starch molecules of the bread. This same type of enzyme will later resume its work in a different organ. In which two locations does this starch-digesting enzyme class actively function? A. Oral cavity and small intestine. B. Stomach and large intestine. C. Oral cavity and stomach. D. Stomach and small intestine. What do you think is the answer? Correct answer, A. Oral cavity and small intestine. Explanation: option A is correct because starch digestion by amylase begins in the oral cavity with salivary amylase and continues in the small intestine with pancreatic amylase, making these the two locations where this enzyme class actively functions. Option B is incorrect because the stomach's acidic environment inactivates salivary amylase, and the large intestine relies on bacterial fermentation rather than amylase enzymes for any remaining carbohydrate breakdown processes. Option C is incorrect because while amylase does begin working in the oral cavity, the highly acidic pH of the stomach inactivates salivary amylase, so the stomach is not a location where this enzyme class actively functions. Option D is incorrect because the stomach does not actively use amylase for starch digestion due to its acidic environment. Starch digestion resumes only when pancreatic amylase is released into the small intestine. The question tells you a patient bites into bread and an enzyme in saliva starts breaking starch bonds immediately. That enzyme is amylase. Then the question asks where this same enzyme class works again later. Starch digestion by amylase happens in two locations: the oral cavity and the small intestine. In the mouth, salivary amylase gets secreted by the parotid and other salivary glands and immediately starts chopping polysaccharides into smaller sugars. But once that food bolus hits the stomach, the extremely acidic environment around pH 2 denatures and inactivates salivary amylase. So the stomach is a dead zone for starch digestion. Amylase picks back up in the small intestine when the pancreas secretes pancreatic amylase into the duodenum. From there, brush border enzymes finish the job. The large intestine doesn't use amylase either. Any remaining carbohydrate fiber gets fermented by gut bacteria, not digested by enzymes. Here's a WGU D313 OA exam tip for you. Amylase works in two places: mouth and small intestine. The stomach kills it, and the large intestine doesn't use it. Quick recall hook. Amylase has two homes: mouth and mini intestine. M&M. 24. A gastroenterologist is explaining protein digestion to a patient with digestive concerns. She describes how protein digestion begins in one organ with a specific enzyme and then continues in the small intestine with a different key enzyme that also activates additional proteases. Which pair of enzymes correctly represents the sequence of protein digestion in these two locations? A. Salivary amylase, then pancreatic lipase. B. Lactase, then pancreatic amylase. C. Pepsin, then trypsin. D. Trypsin, then pepsin. What do you think is the answer? Correct answer, C. Pepsin, then trypsin. Explanation: option C is correct because protein digestion begins in the stomach with pepsin breaking peptide bonds, then continues in the small intestine with trypsin, which further digests proteins and activates additional pancreatic proteases. Option A is incorrect because salivary amylase breaks down starches in the oral cavity, and pancreatic lipase digests fats in the small intestine, and neither enzyme is involved in the process of protein digestion. Option B is incorrect because lactase breaks down the milk. Sugar lactose and pancreatic amylase digests carbohydrates, meaning both enzymes target non-protein substrates and are irrelevant to protein digestion sequences. Option D is incorrect because this reverses the correct anatomical sequence. Pepsin acts first in the stomach, before trypsin continues protein digestion in the small intestine, not the other way around. Protein digestion follows a specific two-step enzyme sequence across two organs. It starts in the stomach with pepsin and continues in the small intestine with trypsin. In the stomach, parietal cells secrete hydrochloric acid that converts pepsinogen from chief cells into pepsin. Pepsin then starts breaking peptide bonds in proteins. That's step 1. When the partially digested proteins reach the small intestine as part of the chyme, the pancreas delivers trypsin through the pancreatic duct. Trypsin continues cutting proteins into smaller peptides, and it also activates other pancreatic proteases, making it a master switch for protein digestion in the small intestine. That's step 2. Salivary amylase and pancreatic lipase target carbs and fats respectively, not proteins. Lactase breaks down lactose, a milk sugar, so that's a carbohydrate enzyme too, and trypsin then pepsin reverses the correct anatomical order. Pepsin always comes first because the stomach comes before the small intestine. Here's a WGU D313 OA exam tip for you. Protein digestion order is pepsin first in the stomach, trypsin second in the small intestine, never the reverse. Quick recall hook, P before T, just like in the alphabet. Pepsin before trypsin, stomach before small intestine. 25. A medical student learns that the cephalic phase of gastric regulation can be triggered before any food physically enters the stomach. Her professor describes how the brain sends preparatory signals through a specific cranial nerve to stimulate early gastric secretions. Which brain regions and cranial nerve initiate this anticipatory digestive response before food reaches the stomach? A. Pons and facial nerve. B. Thalamus and trigeminal nerve. C. Brainstem and glossopharyngeal nerve. D. Cerebral cortex, hypothalamus and vagus nerve. Can you guess the answer? Correct answer, D. Cerebral cortex, hypothalamus and vagus nerve. Explanation. Option D is correct because during the cephalic phase, the cerebral cortex and hypothalamus process sensory cues about food and send signals through the vagus nerve to stimulate gastric secretions before food arrives. Option A is incorrect because while the pons contains salivary nuclei that control salivation, the cephalic phase of gastric regulation specifically involves the cerebral cortex and hypothalamus sending signals through the vagus nerve. Option B is incorrect because the thalamus serves as a sensory relay center and the trigeminal nerve provides facial sensation, but neither is the primary pathway that initiates the anticipatory gastric secretions of the cephalic phase. Option C is incorrect because while the vagus nerve does originate in the brainstem, the cephalic phase specifically requires the cerebral cortex and hypothalamus to process the thought or smell of food before signaling begins. The cephalic phase of gastric regulation is the get ready phase. It happens before food even reaches the stomach. Just thinking about food, smelling it, or tasting it in your mouth is enough to kick things off. The cerebral cortex processes the thought or sight of food. The hypothalamus processes the smell. Together they send signals down the vagus nerve, that's cranial nerve 10, straight to the stomach. The vagus nerve then stimulates gastric glands to start secreting acid, enzymes, and mucus in preparation for incoming food. The key players are the cerebral cortex, the hypothalamus, and the vagus nerve. The pons and facial nerve are involved in salivation, not gastric secretion. The thalamus relays sensory information, but doesn't initiate gastric responses. The glossopharyngeal nerve handles taste from the back of the tongue, but doesn't drive the cephalic phase. Here's a WGU D313 OA exam tip for you. Cephalic phase means head phase. Cephalic literally means head. The brain triggers the stomach before food arrives. Cerebral cortex, hypothalamus, vagus nerve. Quick recall hook. CHV, cortex, hypothalamus, vagus, the cephalic head vibe. 26. A dietitian explains to a patient that one class of macronutrients is synthesized by plants during photosynthesis and serves as the body's preferred source of quick energy by providing glucose for cellular respiration. The patient asks which food groups are the richest dietary sources of this macronutrient class. Which answer best identifies these sources? A. Eggs, ham and soy products. B. Vegetables, fruits, grains and legumes. C. Butter, oils and animal fat. D. Fish, poultry and dairy. Can you guess the answer? Correct answer, B. Vegetables, fruits, grains and legumes. Explanation. Option B is correct because carbohydrates are produced by plants through photosynthesis, and the richest dietary sources include vegetables, fruits, grains and legumes, all of which provide glucose used for cellular energy. Option A is incorrect because eggs, ham and soy products are primarily classified as protein sources that provide amino acids for building enzymes, hormones and structural tissues rather than serving as primary carbohydrate sources. Option C is incorrect because butter, oils and animal fat are lipid sources that provide 9 calories per gram for long-term energy storage, not the plant-derived carbohydrates used as the body's preferred quick fuel. Option D is incorrect because fish, poultry and dairy are predominantly protein and fat sources that supply amino acids and fatty acids, and they are not the primary dietary sources of plant-derived carbohydrates. The question describes a macronutrient made by plants during photosynthesis that serves as the body's preferred quick energy source by providing glucose, that's carbohydrates, and the richest dietary sources are vegetables, fruits, grains and legumes. Carbohydrates are plant-based energy molecules. Plants build them during photosynthesis by converting sunlight, water, and carbon dioxide into glucose and starches. When you eat carb-rich foods, your body breaks those starches and sugars down into glucose, which enters cells and gets converted to ATP through cellular respiration. Vegetables, fruits, grains, and legumes are all plant foods packed with carbohydrates. Eggs, ham, and soy are protein sources. They supply amino acids, not primarily glucose. Butter, oils, and animal fat are lipid sources at 9 calories per gram. Fish, poultry, and dairy are protein and fat sources. None of those are where you go for plant-derived carbohydrates. Here's a WGU D313 OA exam tip for you. Photosynthesis, plants, glucose, and quick energy all point to carbohydrates. Plant-based food groups are the carb sources. Quick recall hook. Carbs come from crops. Plants make carbs, animals make protein. 27. A physician sees a malnourished patient who has been consuming almost no protein for several months. The patient presents with significant loss of skeletal muscle mass. The physician explains that the body has been breaking down its own muscle tissue to obtain specific building blocks it desperately needs. What are these building blocks primarily used to produce in the body? A. Bile salts and bicarbonate ions. B. Glycogen and triglycerides only. C. Glucose and ketone bodies only. D. Enzymes, neurotransmitters, DNA and RNA. Do you know the answer? Correct answer, D. Enzymes, neurotransmitters, DNA and RNA. Explanation. Option D is correct because amino acids obtained from protein breakdown are used to produce enzymes, neurotransmitters, DNA, RNA, receptors, some hormones, ion channels, and contractile muscle proteins essential for body function. Option A is incorrect because bile salts are produced by the liver from cholesterol, and bicarbonate ions are secreted by pancreatic cells. And neither requires amino acids from skeletal muscle catabolism as primary building blocks. Option B is incorrect because glycogen is synthesized from glucose through glycogenesis, and triglycerides are formed through lipogenesis from acetyl-CoA, and neither process requires amino acids as its primary building material. Option C is incorrect, because while amino acids can be converted to glucose through gluconeogenesis during starvation, the primary purpose of dietary protein is building functional molecules like enzymes and neurotransmitters, not just fuel. A malnourished patient with almost no protein intake for months is losing skeletal muscle mass. The body is catabolizing its own muscle tissue to get amino acids. Why? Because amino acids are the building blocks for producing enzymes, neurotransmitters, DNA and RNA. That's the correct answer. Proteins aren't just structural. Amino acids are needed to build the molecules that run virtually every process in the body. Enzymes catalyze metabolic reactions. Neurotransmitters carry signals between neurons. DNA and RNA carry and express genetic information. Amino acids also build receptors, some hormones, ion channels, and contractile muscle fibers. When dietary protein is absent, the body has no choice but to strip amino acids from its own muscle to keep making these critical molecules. Bile salts are made from cholesterol in the liver, not from amino acids. Glycogen is made from glucose. Ketone bodies come from fat breakdown. Those aren't what the body is desperately after when it cannibalizes muscle. Here's a WGU D313 OA exam tip for you. Muscle wasting plus protein deficiency always points to the body needing amino acids for enzymes, neurotransmitters, DNA, and RNA. Quick recall hook: ENDAR enzymes, neurotransmitters, DNA, RNA. That's what amino acids build. 28. A nutrition instructor explains that one macronutrient class provides more than double the calories per gram compared to the other two macronutrient classes. She adds that this same nutrient class also cushions organs, insulates neurons, stores certain vitamins, and serves as a precursor to some hormones. Which macronutrient class and caloric value is she describing? A. Lipids at 9 calories per gram. B. Proteins at 9 calories per gram. C. Carbohydrates at 9 calories per gram. D. Lipids at 4 calories per gram. Can you guess the answer? Correct answer: A. Lipids at 9 calories per gram. Explanation: Option A is correct because lipids provide 9 calories per gram, which is more than double the 4 calories per gram from carbohydrates and proteins, and lipids cushion organs, insulate neurons, and store fat-soluble vitamins. Option B is incorrect because proteins provide only 4 calories per gram, not 9, and while proteins serve many structural and enzymatic functions in the body, they are not the primary macronutrient for cushioning organs. Option C is incorrect because carbohydrates provide 4 calories per gram, not 9, and their primary role is supplying glucose for quick energy through cellular respiration rather than cushioning organs or insulating neurons. Option D is incorrect because although lipids do perform the functions described in the question, including cushioning and insulation, the caloric value stated is wrong. Lipids provide 9 calories per gram, not 4. The question describes a macronutrient that provides more than double the calories per gram compared to the other two. It also cushions organs, insulates neurons, stores fat-soluble vitamins, and serves as a hormone precursor. That's lipids at 9 calories per gram. Carbohydrates and proteins both provide 4 calories per gram. Lipids blow past them at 9, more than double. That caloric density is why fat is the body's preferred long-term energy storage molecule. But lipids do far more than store energy. They cushion and protect vital organs from physical impact. They form the myelin sheath that insulates neurons for fast signal transmission. They store fat-soluble vitamins A, D, E, and K. And they serve as precursors for steroid hormones like estrogen and testosterone. Proteins at 9 calories is wrong. Proteins are 4. Carbohydrates at 9 is wrong, also 4. Lipids at 4 calories is wrong on the number, even though the functions described are correct. Here's a WGU D313 OA exam tip for you. 9 calories per gram plus cushion, insulate, and hormone precursor equals lipids. No other macronutrient hits all those marks. Quick recall hook. Lipids equals 9. LIPIDS has more letters than CARBS. More letters, more calories. 29. A gastroenterologist recommends that a patient with slow digestive transit increase her intake of insoluble fibre. The patient asks why this type of carbohydrate is beneficial even though the human body cannot actually digest it. Which explanation best describes why insoluble fibre helps promote healthy digestive function? A. It provides glucose for cellular respiration. B. It dissolves food for taste receptor detection. C. It increases stool bulk and reduces transit time. D. It emulsifies fats for better lipid absorption. What do you think is the answer? Correct answer. C. It increases stool bulk and reduces transit time. Explanation. Option C is correct because insoluble fibre cannot be digested by humans but increases stool bulk, which reduces the transit time of food through the digestive tract and promotes regular, healthy bowel movements. Option A is incorrect because insoluble fibre cannot be broken down by human digestive enzymes into glucose, so it does not contribute to cellular respiration or provide usable energy for the body's metabolic needs. Option B is incorrect because dissolving food molecules so taste receptors can detect them is a function of saliva in the oral cavity, not a benefit provided by insoluble fibre in the digestive tract. Option D is incorrect because fat emulsification is performed by bile produced by the liver and stored in the gallbladder, not by insoluble fibre, which has no role in the chemical processing of dietary lipids. The question asks why insoluble fibre is beneficial even though we can't digest it. The answer is straightforward. It increases stool bulk and reduces transit time. Insoluble fibre is a type of carbohydrate that human digestive enzymes simply cannot break down. It passes through the GI tract basically intact, but that's exactly why it's useful. As it travels through the large intestine, it absorbs water and adds bulk to the stool. Bulkier stool stimulates the intestinal walls more effectively, which triggers stronger peristaltic contractions and mass movements. The result is faster transit time. Food residue moves through the colon quicker, and you have more regular bowel movements. It doesn't provide glucose because we can't digest it into absorbable sugars. Dissolving food for taste receptors is what saliva does in the mouth. Emulsifying fats is bile's job. None of those apply to fibre. Here's a WGU D313 OA exam tip for you. Insoluble fibre equals bulk equals faster transit equals regular bowel movements. If the question says can't digest but still helpful, think fibre and stool bulk. Quick recall hook. Fibre is the fast lane. It speeds things up by adding bulk. 30. A nutrition student learns that amino acids fall into 2 categories, based on whether the body can manufacture them internally. Her professor explains that one category must be obtained entirely from dietary sources because the body lacks the biochemical pathways to create them. Which statement correctly distinguishes these 2 amino acid categories? A. Both categories are synthesized within the body. B. Essential amino acids are made by the liver. C. Non-essential amino acids must come from food. D. Essential amino acids must come from food. What do you think is the answer? Correct answer. D. Essential amino acids must come from food. Explanation. Option D is correct because essential amino acids cannot be synthesized by the human body and must be obtained entirely from dietary sources, while non-essential amino acids can be produced internally through transamination reactions. Option A is incorrect because only non-essential amino acids can be synthesized within the body. Essential amino acids lack the necessary biosynthetic pathways and must be consumed through protein-containing foods in the diet. Option B is incorrect because essential amino acids cannot be manufactured by the liver or any other organ in the body, which is precisely why they are classified as essential and must come from dietary intake. Option C is incorrect because this reverses the definitions. Non-essential amino acids can be synthesized within the body through metabolic reactions, so they do not need to come exclusively from food sources. This question tests whether you know the difference between essential and non-essential amino acids. The answer is that essential amino acids must come from food. Here's the distinction. Your body can manufacture non-essential amino acids on its own through transamination reactions. The liver shuffles amino groups between molecules to build the ones it needs. But essential amino acids lack those biosynthetic pathways entirely. The body cannot make them no matter what. If you don't eat them, you don't have them. That's what makes them essential to your diet. Animal protein sources like meat, eggs, and dairy contain all the essential amino acids in one package, which is why they're called complete proteins. Most plant protein sources are missing one or more essential amino acids, so vegetarians need to combine different plant sources to get the full set. The wrong answers either flip the definitions, claim both categories are made internally, or say the liver makes essential amino acids. None of that is true. Here's a WGU D313 OA exam tip for you. Essential means you essentially must eat it. If the body could make it, it wouldn't be essential. Quick recall hook. Essential means eat it. If you don't eat it, you don't have it. 31. After eating a large meal, a patient's blood glucose rises significantly. Her endocrinologist explains that a specific hormone becomes dominant during this fed state and works to move glucose from the bloodstream into cells for energy production and storage. Which hormone dominates during this absorptive state and which cells produce it? A. Insulin from pancreatic beta cells. B. Glucagon from pancreatic alpha cells. C. Gastrin from stomach G cells. D. Secretin from duodenal cells. Can you guess the answer? Correct answer, A. Insulin from pancreatic beta cells. Explanation. Option A is correct because insulin is the dominant hormone during the absorptive state, produced by beta cells in the pancreatic islets, and it acts to move glucose from the blood into cells for energy, use, or storage. Option B is incorrect because glucagon is produced by alpha cells and is the dominant hormone during the post-absorptive fasting state, when it mobilizes stored glucose into the bloodstream rather than storing it in cells. Option C is incorrect because gastrin is a digestive hormone produced by G cells in the stomach that stimulates acid secretion and stomach contractions, and it does not regulate blood glucose movement into body cells. Option D is incorrect because secretin is released by the duodenum to stimulate pancreatic bicarbonate secretion for neutralizing acidic chyme, and it plays no direct role in regulating blood glucose levels during the absorptive state. After a large meal, blood glucose spikes. The question asks which hormone takes charge during this absorptive state. The answer is insulin from pancreatic beta cells. The absorptive state is just a technical name for the fed state, the period after eating when nutrients from your meal are flooding the bloodstream. During this state, insulin is king. Beta cells in the islets of Langerhans detect the rising blood glucose and respond by dumping insulin into the blood. Insulin then does its main job. It signals cells throughout the body to open their glucose transporters and pull glucose out of the blood. That glucose gets used immediately for ATP production, or stored as glycogen in the liver and muscles, or converted to fat for long-term storage. Glucagon from alpha cells is the opposite player. It dominates during fasting, mobilizing stored glucose back into the blood. Gastrin is a digestive hormone for stomach acid, not glucose regulation. Secretin triggers bicarbonate release from the pancreas for neutralizing chyme. Here's a WGU D313 OA exam tip for you. Fed state after eating, absorptive, blood glucose high. All of those are code for insulin from beta cells. Quick recall hook. Insulin moves glucose into cells. Insulin gets glucose in. 32. Between meals, a patient's blood glucose begins to drop. Her body responds by breaking down a stored polysaccharide, primarily found in the liver, and muscles to release glucose back into the bloodstream and maintain normal levels. Which metabolic process describes this specific breakdown of stored polysaccharide to release glucose? A. Lipogenesis. B. Gluconeogenesis. C. Glycogenolysis. D. Beta oxidation. Do you know the answer? Correct answer C. Glycogenolysis. Explanation. Option C is correct because glycogenolysis is the catabolic reaction that breaks down stored glycogen, primarily in the liver and muscles, and releases glucose into the bloodstream to maintain blood glucose between meals. Option A is incorrect because lipogenesis is the anabolic process of synthesizing triglycerides from acetyl-CoA for long-term fat storage, which is the opposite of breaking down stored molecules to release glucose into the blood. Option B is incorrect because gluconeogenesis is the synthesis of entirely new glucose molecules from non-sugar precursors like pyruvate, lactate, and amino acids, not the breakdown of already stored glycogen reserves. Option D is incorrect because beta oxidation is the process of breaking down fatty acid chains into acetyl-CoA units within the mitochondria for energy production, and it does not directly release glucose into the bloodstream. Between meals, blood glucose starts dropping. The body needs to top it back up, and the first thing it reaches for is glycogen, a stored polysaccharide packed away in the liver and muscles. The process of breaking down glycogen to release glucose is called glycogenolysis. Break the word apart, and it tells you everything. Glycogenolysis. Glycogen is the stored sugar. Lysis means to break apart, so glycogenolysis literally means breaking apart glycogen. This is a catabolic reaction. It tears a big molecule into smaller pieces. The glucose released from glycogen goes straight into the bloodstream to keep blood sugar levels stable between meals. Lipogenesis is building fat, not breaking glycogen. Gluconeogenesis is making brand new glucose from non-sugar sources like pyruvate and amino acids. That's different because it builds glucose from scratch rather than releasing it from storage. Beta oxidation breaks down fatty acids, not glycogen. Here's a WGU D313 OA exam tip for you. Glycogenolysis equals glycogen breakdown. If the question says stored polysaccharide and releasing glucose between meals, that's glycogenolysis. Quick recall hook. Lysis means break. Glycogenolysis breaks glycogen. The suffix tells you the action. 33. A physician explains to a fasting patient that once glycogen stores are depleted, the body can still manufacture glucose from molecules that are not carbohydrates. She lists pyruvate, lactate, glycerol, and certain amino acids as possible starting materials for this glucose-building process. Which metabolic process uses these non-sugar precursors to synthesize new glucose? A. Glycolysis. B. Gluconeogenesis. C. Lipogenesis. D. Glycogenolysis. Do you know the answer? Yes. Correct answer, B. Gluconeogenesis. Explanation. Option B is correct because gluconeogenesis is the metabolic process that synthesizes new glucose molecules from non-sugar precursors, including pyruvate, lactate, glycerol, and the amino acids alanine and glutamine during fasting conditions. Option A is incorrect because glycolysis is the metabolic pathway that oxidizes existing glucose molecules to produce ATP for cellular energy, which is the breakdown of glucose rather than the creation of new glucose. Option C is incorrect because lipogenesis is the process of forming triglycerides from acetyl-CoA for fat storage in adipose tissue, and it builds lipid molecules rather than synthesizing new glucose from non-sugar sources. Option D is incorrect. because glycogenolysis breaks down previously stored glycogen to release glucose. But the question describes a situation where glycogen stores are already depleted and new glucose must be built from scratch. This question describes a scenario where glycogen is already gone and the body still needs glucose. So it builds new glucose from non-carbohydrate sources: pyruvate, lactate, glycerol, and certain amino acids. That process is gluconeogenesis. Break it down. Gluco means glucose, neo means new, genesis means creation. So gluconeogenesis means creating new glucose. This kicks in during extended fasting when glycogen stores have been used up. The liver takes non-sugar molecules: pyruvate from glycolysis, lactate from anaerobic metabolism, glycerol from fat breakdown, and the amino acids alanine and glutamine from protein, and chemically converts them into glucose. Glycolysis is the exact opposite. It breaks glucose down for energy. It doesn't build it. Lipogenesis builds fat from acetyl-CoA for storage, not glucose. Glycogenolysis breaks down stored glycogen, but the question says glycogen is already depleted. Here's a WGU D313 OA exam tip for you. New glucose from non-sugar sources is the definition of gluconeogenesis. If you see pyruvate, lactate, glycerol, or amino acids being turned into glucose, that's it. Quick recall hook. Neo means new. Gluconeogenesis means making new glucose, fresh glucose from scratch. 34. A critical care physician is treating a patient who has not eaten for several days. She explains to her medical team the predictable sequence in which the body consumes its own fuel reserves during prolonged starvation. Which sequence correctly describes the order in which energy sources are depleted during extended fasting? A. Fat stores first, then glycogen, then protein. B. Protein first, then fat stores, then glycogen. C. Glycogen first, then protein, then fat stores. D. Glycogen first, then fat stores, then protein. What do you think is the answer? Correct answer: D. Glycogen first, then fat stores, then protein. Explanation. Option D is correct because during starvation, the body first depletes glycogen reserves within roughly 12 to 24 hours, then mobilizes fat stores producing ketone bodies, and finally catabolizes muscle protein as a last resort. Option A is incorrect because fat stores are not the first fuel source used during starvation. The body initially relies on glycogen reserves in the liver and muscles before transitioning to fat mobilization and ketone body production. Option B is incorrect because protein from muscle tissue is the last energy source the body uses during starvation, not the first, since catabolizing muscle causes severe functional damage and organ compromise over time. Option C is incorrect because after glycogen depletion, the body shifts to fat stores and ketone body production rather than immediately catabolizing muscle protein, which is preserved as the final emergency fuel reserve. During starvation, the body burns through its fuel reserves in a specific, predictable order. It goes glycogen first, then fat stores, then protein. Glycogen is the quickest fuel to access. It's stored in the liver and muscles and can be broken down into glucose within hours. But glycogen reserves are limited. They're usually gone within 12 to 24 hours. Once glycogen runs out, the body shifts to fat stores. Fat breakdown through lipolysis and beta-oxidation releases fatty acids and produces ketone bodies, which can fuel the brain and heart. Fat stores can last weeks or even months, depending on body composition. Protein from muscle tissue is the absolute last resort. The body avoids it as long as possible because catabolizing muscle damages organ function and impairs movement. Only when fat reserves are severely depleted does the body start consuming its own muscle protein for energy. This is why prolonged starvation causes visible muscle wasting and eventually organ failure. Here's a WGU D313 OA exam tip for you. Starvation fuel order is GFP: glycogen, fat, protein. Always in that order, never rearranged. Quick recall hook. GFP: Go for protein last. The body protects muscle as long as it can. 35. During prolonged fasting, a patient's liver begins producing alternative fuel molecules from excess acetyl-CoA, generated by rapid fat breakdown. A neurologist explains that the brain can use these molecules as an energy substitute when glucose becomes scarce. Which type of molecule is the liver producing as this alternative brain fuel? A. Ketone bodies. B. Glycogen. C. Pepsinogen. D. Bile salts. Do you know the answer? Correct answer, A. Ketone bodies. Explanation: option A is correct because during prolonged fasting, the liver produces ketone bodies from excess acetyl-CoA, generated by rapid fat breakdown. And the brain can use these molecules as an alternative energy source when glucose is scarce. Option B is incorrect because glycogen is a storage form of glucose found in the liver and muscles. And during prolonged fasting, glycogen stores have already been depleted rather than being newly produced by the liver. Option C is incorrect because pepsinogen is an inactive digestive enzyme precursor secreted by chief cells in the stomach for protein digestion, and it has no role as an alternative fuel source for the brain. Option D is incorrect because bile salts are components of bile produced by the liver to emulsify dietary fats in the duodenum, and they serve a digestive function rather than acting as an alternative energy source. During prolonged fasting, fat gets broken down rapidly. Beta oxidation chops fatty acid chains into acetyl-CoA units. When there's more acetyl-CoA than the citric acid cycle can handle, the liver converts the excess into ketone bodies. Acetoacetate is one example. Ketone bodies are water-soluble molecules that can travel through the blood to tissues that need fuel. The brain is the big deal here. Normally the brain runs almost entirely on glucose because fatty acids can't cross the blood-brain barrier, but ketone bodies can. So during extended fasting or starvation, ketone bodies become the brain's backup fuel source. The heart also uses ketone bodies heavily after several days without food. Glycogen would be wrong because glycogen stores are already gone by the time the liver starts cranking out ketone bodies. Pepsinogen is a stomach enzyme for digesting protein, completely unrelated to fasting metabolism. Bile salts emulsify fat in the duodenum, also unrelated to alternative brain fuel. Here's a WGU D313 OA exam tip for you. Excess acetyl-CoA from fat breakdown plus alternative brain fuel equals ketone bodies. If the question mentions the brain using something other than glucose during fasting, it's ketone bodies. Quick recall hook. Ketones keep the brain going. K for ketones, K for keep alive. 36. A cardiologist is teaching a student to identify heart sounds using a stethoscope. She explains that the first heart sound heard during each cardiac cycle is produced when specific valves close as the ventricles begin contracting. The student asks which valves produce this characteristic first sound. Which pair of valves is responsible for the first heart sound? A. Aortic and pulmonary valves. B. Aortic and tricuspid valves. C. Mitral and tricuspid valves. D. Mitral and pulmonary valves. What do you think is the answer? Valves. Correct answer, C. Mitral and tricuspid valves. Explanation: option C is correct, because the first heart sound is produced by the closing of the two atrioventricular valves, specifically the mitral valve on the left side and the tricuspid valve on the right side, as ventricular systole begins. Option A is incorrect because the aortic and pulmonary valves are the semilunar valves whose closing produces the second heart sound, not the first, occurring at the end of ventricular systole rather than the beginning. Option B is incorrect because while the tricuspid valve does contribute to the first heart sound, the aortic valve is a semilunar valve that closes during the second heart sound, making this an incorrectly mixed pairing. Option D is incorrect because while the mitral valve does contribute to the first heart sound, The pulmonary valve is a semilunar valve that closes during the second heart sound, making this combination an incorrect mixed pairing. Heart sounds come from valves closing, and you need to know which valves make which sound. The first heart sound, S1, the lub, is produced by the closing of the two atrioventricular valves. Those are the mitral valve on the left and the tricuspid valve on the right. These valves sit between the atria and the ventricles. When the ventricles start contracting during systole, the pressure pushes blood upward against these valves and slams them shut. That slam is S1. The second heart sound, S2, the dub, comes from the semilunar valves closing, the aortic valve and the pulmonary valve, those close at the end of ventricular systole when the ventricles relax and blood tries to fall back. The wrong answers mix AV valves with semilunar valves. If you see aortic or pulmonary paired with anything, that's S2 territory, not S1. The mitral valve is also called the bicuspid valve, two names for the same valve on the left side. Here's a WGU D313 OA exam tip for you. S1 is the AV valves, mitral and tricuspid. S2 is the semilunar valves, aortic and pulmonary. Quick recall hook, MT for S1, mitral tricuspid. Think my turn, MT. The AV valves close first. 37. A pulmonologist explains to a medical student that carbon dioxide is transported in the blood through multiple mechanisms. She describes how an enzyme inside red blood cells catalyzes a reaction that produces hydrogen ions and bicarbonate ions from carbonic acid. Which enzyme performs this critical catalytic function inside the red blood cells? A. Pepsin. B. Salivary amylase. C. Pancreatic lipase. D. Carbonic anhydrase. Can you guess the answer? Correct answer, D. Carbonic anhydrase. Explanation. Option D is correct because carbonic anhydrase is the enzyme inside red blood cells that catalyzes the dissociation of carbonic acid into hydrogen ions and bicarbonate ions, enabling efficient carbon dioxide transport in the blood. Option A is incorrect because pepsin is a protein-digesting enzyme that functions in the acidic environment of the stomach, and it has no role in catalyzing carbon dioxide transport reactions within red blood cells. Option B is incorrect because salivary amylase is a carbohydrate digesting enzyme found in saliva that breaks down starches in the oral cavity, and it is not present in red blood cells or involved in gas transport. Option C is incorrect because pancreatic lipase is a fat digesting enzyme secreted into the duodenum to break down triglycerides, and it plays no role in the enzymatic reactions related to carbon dioxide transport in blood. Carbon dioxide transport in the blood depends heavily on one enzyme inside red blood cells, carbonic anhydrase. Here's the process. CO2 enters a red blood cell from the tissues. Inside the cell, carbonic anhydrase catalyzes the reaction, where CO2 combines with water to form carbonic acid. Then carbonic anhydrase catalyzes the next step too. It splits that carbonic acid into hydrogen ions and bicarbonate ions. The bicarbonate ions then get shuttled out of the red blood cell into the plasma, where they become the main form of CO2 transport in the blood. This is the most common way carbon dioxide travels, not dissolved in plasma and not bound to hemoglobin, though both of those happen too. Pepsin is a protein-digesting enzyme in the stomach. Salivary amylase breaks starch in the mouth. Pancreatic lipase breaks fat in the duodenum. None of those are anywhere near red blood cells or involved in gas transport. Here's a WGU D313 OA exam tip for you. The enzyme inside red blood cells that handles CO2 transport is always carbonic anhydrase. If you see CO2, bicarbonate, or hydrogen ions inside red blood cells, that's your answer. Quick recall hook. Carbonic anhydrase handles carbon dioxide, Ca for CO2. 38. As carbon dioxide levels in the blood increase, bicarbonate ions accumulate inside red blood cells and then move out into the plasma along their concentration gradient. To maintain electrical balance across the cell membrane, another ion must simultaneously move in the opposite direction. Which ion moves into the red blood cell to maintain this electrical neutrality? A. Chloride, B. Sodium, C. Potassium, D. Calcium. Can you guess the answer? Correct answer, A. Chloride. Explanation: option A is correct because during the chloride shift, a chloride ion moves into the red blood cell from the plasma for every bicarbonate ion that moves out, maintaining the electrical balance across the cell membrane. Option B is incorrect because sodium ions are primarily involved in nerve impulse transmission and fluid balance regulation through the sodium-potassium pump, and they do not participate in the chloride shift exchange mechanism. Option C is incorrect because potassium ions function mainly in maintaining resting membrane potential and cardiac muscle contraction, and they are not the ion exchanged for bicarbonate during the chloride shift process. Option D is incorrect because calcium ions are involved in muscle contraction, blood clotting, and bone formation, and they do not participate in the ion exchange mechanism that maintains electrical balance during bicarbonate transport. This question is about the chloride shift, a specific ion exchange that happens during CO2 transport. Here's the setup. Inside red blood cells, carbonic anhydrase splits carbonic acid into bicarbonate ions and hydrogen ions. Bicarbonate ions accumulate inside the cell. Eventually, the concentration gradient pushes bicarbonate ions out of the red blood cell and into the plasma. But every time a negatively charged bicarbonate ion leaves, the cell would become electrically unbalanced. To fix that, a chloride ion, also negatively charged, moves into the red blood cell from the plasma. One chloride in for every bicarbonate out. That's the chloride shift. It maintains electrical neutrality across the red blood cell membrane. Sodium is involved in nerve impulses and the sodium-potassium pump. Potassium maintains resting membrane potential. Calcium handles muscle contraction and blood clotting. None of those participate in this specific bicarbonate exchange. Here's a WGU D313 OA exam tip for you. Bicarbonate leaves the red blood cell, chloride enters, 1 for 1. That's the chloride shift. If you see bicarbonate moving out and a question about what moves in, the answer is chloride. Quick recall hook, chloride shift. Chloride comes in as bicarbonate goes out. C replaces C. 39. An exercise physiologist explains that during intense physical activity, working muscles produce heat and acidic byproducts. These local tissue conditions cause hemoglobin to release oxygen more readily at the sites where it is most needed. Which two specific changes in the tissue environment promote this increased oxygen release from hemoglobin? A. Lower temperature and higher pH. B. Higher temperature and lower pH. C. Lower temperature and lower pH. D. Higher temperature and higher pH. Can you guess the answer? Correct answer. B. Higher temperature and lower pH. Explanation: option B is correct because higher temperature and lower pH in active muscle tissue promote the dissociation of oxygen from hemoglobin, ensuring oxygen is released where metabolic demand is greatest, as described by the Bohr effect. Option A is incorrect because lower temperature and higher pH would actually increase hemoglobin's affinity for oxygen, causing it to hold on to oxygen more tightly rather than releasing it to the working muscle tissues. Option C is incorrect because while lower pH does promote oxygen release from hemoglobin, lower temperature would counteract this by increasing oxygen binding affinity, and active muscles generate heat rather than cooling down. Option D is incorrect because while higher temperature does promote oxygen release, higher pH would increase hemoglobin's affinity for oxygen and work against the release mechanism, creating contradictory effects on oxygen delivery. When muscles work hard, two things happen locally. Temperature goes up and pH goes down. Muscle contractions generate heat, and the lactic acid and CO2 produced by rapid metabolism make the tissue more acidic. These two changes, higher temperature and lower pH, cause hemoglobin to release oxygen more readily. This is the Bohr effect. Hemoglobin's affinity for oxygen drops when the environment gets hotter and more acidic, which is exactly what active muscles need. Where oxygen demand is highest, the conditions automatically force hemoglobin to let go of its oxygen. It's an elegant delivery system. Oxygen gets released precisely where it's needed most. Lower temperature and higher pH would do the opposite. Hemoglobin would grip oxygen tighter and not release it. Lower temperature with lower pH sends a mixed signal. Temperature working against release. Higher temperature with higher pH is also mixed. Only higher temperature combined with lower pH consistently drives oxygen release. Here's a WGU D313 OA exam tip for you. Hot and acidic equals oxygen release. The Bohr effect means hemoglobin drops oxygen in harsh conditions. Quick recall hook. Hot and hostile makes HB let go. Higher temp, lower pH, oxygen delivered. 40. A hematologist explains to a patient that one type of blood cell contains an iron-rich protein specifically designed to bind and transport oxygen molecules from the lungs to tissues throughout the body. This same cell type holds the highest concentration of iron of any cell in the human body. Which blood cell type is the hematologist describing? A. Monocytes, B. Lymphocytes, C. Platelets, D. Erythrocytes. What do you think is the answer? Correct answer, D. Erythrocytes. Explanation: option D is correct because erythrocytes are red blood cells that contain hemoglobin, an iron-rich protein that binds and transports oxygen, and they hold the highest concentration of iron of any cell in the body. Option A is incorrect because monocytes are a type of agranulocyte white blood cell that functions in immune defense through phagocytosis, and they do not contain hemoglobin or serve as the primary oxygen transport cells. Option B is incorrect because lymphocytes are agranulocyte white blood cells involved in adaptive immune responses, including antibody production and cell-mediated immunity, and they do not transport oxygen using hemoglobin. Option C is incorrect because platelets are small cell fragments involved in blood clotting and the formation of platelet plugs at wound sites, and they do not contain the iron-rich hemoglobin needed for oxygen transport. The question describes a blood cell containing an iron-rich protein that binds and transports oxygen, and says this cell type has the highest iron concentration in the body. That's erythrocytes, red blood cells. Erythrocytes are packed with hemoglobin, a protein built around iron-containing heme groups. Each hemoglobin molecule has 4 heme groups, and each heme group can bind one oxygen molecule. That means a single hemoglobin molecule carries up to 4 oxygen molecules. Erythrocytes are essentially sacs of hemoglobin. They've lost their nucleus and most organelles just to make room for more of it. That's why they have the highest iron concentration of any cell. Monocytes are white blood cells that do phagocytosis. They eat pathogens, not carry oxygen. Lymphocytes are white blood cells involved in adaptive immunity. B cells make antibodies and T cells coordinate immune responses. Platelets are cell fragments for blood clotting. None of those contain hemoglobin. Here's a WGU D313 OA exam tip for you. Iron, hemoglobin and oxygen transport all point to erythrocytes. If you see any combination of those 3 terms, the answer is red blood cells. Quick recall hook. Erythro means red. Erythrocytes are red because of iron in hemoglobin. Red equals iron equals oxygen. 41. A pulmonologist explains to a patient how normal inhalation works. She describes how the primary breathing muscle contracts and flattens, which increases the volume of the chest cavity. According to a fundamental gas law, this volume increase causes a pressure change that draws air into the lungs. Which muscle and pressure relationship is she describing? A. External intercostals cause pressure increase. B. Diaphragm contraction causes pressure increase. C. Diaphragm contraction causes pressure decrease. D. Abdominal muscles cause pressure decrease. What do you think is the answer? Correct answer, C. Diaphragm contraction causes pressure decrease. Explanation. Option C is correct because during normal inhalation, the diaphragm contracts and flattens, increasing thoracic cavity volume, which according to Boyle's law decreases pressure and creates a gradient that draws air into the lungs. Option A is incorrect because while external intercostals can assist with breathing during forced inhalation, the primary muscle of respiration is the diaphragm, and volume increase causes pressure to decrease rather than increase. Option B is incorrect because although the diaphragm does contract during inhalation, the resulting increase in thoracic volume causes intrapulmonary pressure to decrease, not increase, which is what draws air into the lungs. Option D is incorrect because abdominal muscles are primarily involved in forced exhalation by compressing the abdominal cavity and pushing the diaphragm upward, and they are not the primary muscles driving normal inhalation. This question is about the mechanics of breathing, specifically what happens during normal, quiet inhalation. The primary muscle of respiration is the diaphragm, a dome-shaped sheet of muscle sitting at the base of the thoracic cavity. When you inhale, the diaphragm contracts and flattens downward. That flattening increases the volume of the thoracic cavity. Now here's where Boyle's law comes in. When volume increases at a constant temperature, pressure decreases. So as the thoracic cavity expands, the pressure inside the lungs drops below atmospheric pressure. Air always flows from high pressure to low pressure, so air rushes into the lungs to equalize the difference. That's inhalation. The diaphragm contracts, volume goes up, pressure goes down, air comes in. External intercostals can assist during forced breathing, but they're not the primary muscle. The diaphragm is. Option B says diaphragm contraction causes pressure increase, which is backward. Abdominal muscles are for forced exhalation, pushing the diaphragm back up. Here's a WGU D313 OA exam tip for you. Diaphragm contracts, volume up, pressure down, air in. That sequence is Boyle's law applied to breathing. Quick recall hook: diaphragm down, pressure down, air drawn in, 3 Ds for inhalation. 42. A critical care physician explains that specialized receptor structures in the body constantly monitor blood gas levels and pH. When carbon dioxide levels rise or oxygen levels fall, these structures send signals to the brainstem to adjust breathing rate and depth. Which specific structure monitors these partial pressures and pH changes in arterial blood? A. Aortic body. B. Pyloric sphincter. C. Parietal cells. D. Ileocecal valve. What do you think is the answer? Correct answer, A. Aortic body. Explanation: option A is correct because the aortic body monitors the partial pressures of carbon dioxide and oxygen, along with pH in the blood, and it sends signals to the brainstem to adjust the rate and depth of breathing accordingly. Option B is incorrect because the pyloric sphincter is a ring of smooth muscle in the stomach that regulates the release of chyme into the duodenum, and it has no sensory function related to monitoring blood gas levels. Option C is incorrect because parietal cells are specialized cells in the gastric glands that secrete hydrochloric acid, an intrinsic factor for digestion, and they do not monitor blood oxygen, carbon dioxide, or pH levels. Option D is incorrect because the ileocecal valve guards the opening between the ileum and the large intestine to prevent backflow, and it has no chemoreceptor function related to respiratory regulation of blood gases. The body needs to constantly monitor blood gas levels to keep breathing on track. The structures responsible for this are chemoreceptors called the aortic bodies and carotid bodies. This question asks specifically about the aortic body. The aortic body sits near the aortic arch and monitors 3 things in the arterial blood: the partial pressure of carbon dioxide, the partial pressure of oxygen, and pH. When CO2 rises, or O2 falls, or pH drops, the aortic body fires signals through nerves to the brainstem's respiratory centres. The brainstem then adjusts breathing rate and depth to correct the imbalance. Faster and deeper breaths blow off more CO2 and bring in more O2. The pyloric sphincter is a muscular valve between the stomach and duodenum. It controls food passage, not blood gases. Parietal cells make HCl and intrinsic factor in the stomach. The ileocecal valve prevents backflow from the large intestine into the small intestine. None of those have anything to do with respiratory monitoring. Here's a WGU D313 OA exam tip for you. Blood gas monitoring, CO2, O2, pH, and breathing adjustments all point to the aortic body or carotid body. The other three options are all digestive structures thrown in as distractors. Quick recall hook. Aortic body monitors the aorta's blood. A for aortic, A for arterial blood monitoring. 43. A nephrologist explains to a medical student that one specific segment of the nephron is primarily responsible for reabsorbing the majority of filtered nutrients, including glucose, amino acids, and vitamins back into the blood before they are lost in the urine. Which nephron segment performs this critical bulk reabsorption of filtered nutrients? A. Collecting duct. B. Loop of Henle. C. Glomerulus. D. Proximal convoluted tubule. What do you think is the answer? Correct answer, D. Proximal convoluted tubule. Explanation: option D is correct because the proximal convoluted tubule reabsorbs the majority of filtered nutrients, including glucose, amino acids, and vitamins from the filtrate back into the blood, preventing their loss in urine. Option A is incorrect because the collecting duct is the final segment, where antidiuretic hormone acts to regulate water reabsorption, and where ammonium ions are secreted for acid-base balance, not where bulk nutrient reabsorption occurs. Option B is incorrect because the loop of Henle is primarily responsible for reabsorbing bicarbonate ions and creating a concentration gradient that allows urine to become more concentrated through the countercurrent multiplier system. Option C is incorrect because the glomerulus is the capillary network, where blood is initially filtered under pressure to produce filtrate, but it does not selectively reabsorb nutrients back into the bloodstream from that filtrate. The nephron is the functional unit of the kidney, and each segment has a different job. The question asks which segment handles bulk reabsorption of filtered nutrients, glucose, amino acids, and vitamins. That's the proximal convoluted tubule, or PCT. When blood gets filtered at the glomerulus, the filtrate contains everything small enough to pass through: water, salts, glucose, amino acids, vitamins, urea, and more. You don't want to lose glucose and amino acids in urine, so the PCT grabs most of it back before the filtrate goes any further. The PCT reabsorbs roughly 65% of all filtered sodium and water, and nearly 100% of filtered glucose and amino acids under normal conditions. It's the first stop after filtration, and it handles the biggest reabsorption workload. The collecting duct is the final segment, where ADH controls water reabsorption. The loop of Henle creates the concentration gradient for producing concentrated urine. The glomerulus does the initial filtering, but doesn't reabsorb anything. It's a one-way sieve. Here's a WGU D313 OA exam tip for you. Bulk nutrient reabsorption. Glucose, amino acids, vitamins, always means the proximal convoluted tubule. Quick recall hook. PCT means primary collection of treasures. It grabs back the good stuff before it's lost. 44. A patient is producing large volumes of very dilute urine despite being dehydrated. Her endocrinologist suspects a deficiency of a specific hormone released by the pituitary gland that normally acts on the collecting ducts to increase water reabsorption and produce more concentrated urine. Which hormone is most likely deficient in this patient? A. Glucagon. B. Antidiuretic hormone C. Gastrin, D. Insulin. Do you know the answer? Correct answer, B. Antidiuretic hormone. Explanation: option B is correct because antidiuretic hormone is released by the pituitary gland and acts on the collecting ducts to increase water reabsorption through aquaporin channels, producing more concentrated urine when the body needs water. Option A is incorrect because glucagon is released by alpha cells of the pancreas to mobilize stored glucose during fasting states, and it has no direct role in regulating water reabsorption in the kidney collecting ducts. Option C is incorrect because gastrin is a digestive hormone released by G cells in the stomach that stimulates hydrochloric acid production and stomach contractions, and it does not affect water reabsorption in the kidneys. Option D is incorrect because insulin is released by beta cells of the pancreas to move glucose from the blood into cells during the absorptive state, and it does not regulate water reabsorption or urine concentration in the kidneys. A dehydrated patient producing large volumes of dilute urine. That's a textbook sign of antidiuretic hormone deficiency. Normally, when the body is dehydrated, the pituitary gland releases ADH into the bloodstream. ADH travels to the kidneys and acts specifically on the collecting ducts. It triggers the insertion of aquaporin channels into the collecting duct cell membranes. Aquaporins are water channels. Once they're in place, water flows from the filtrate back into the blood by osmosis. This concentrates the urine and conserves water for the body. Without ADH, those aquaporin channels don't get inserted. Water stays in the filtrate and gets excreted as dilute urine. The patient loses water they desperately need, which is why they're dehydrated despite producing lots of urine. This condition is called diabetes insipidus. Glucagon mobilizes glucose during fasting. It's a metabolic hormone, not a kidney hormone. Gastrin stimulates stomach acid. Insulin moves glucose into cells. None of those regulate water reabsorption in the kidneys. Here's a WGU D313 OA exam tip for you. Dilute urine plus dehydration plus pituitary gland plus collecting ducts equals ADH deficiency. Quick recall hook. Antidiuretic means anti-peeing. ADH stops you from peeing out all your water. No ADH, too much dilute urine. 45. A renal physiologist describes how urine becomes progressively more concentrated as filtrate passes through a specific looping structure in the nephron. This structure uses a countercurrent multiplier system to create an osmotic gradient in the surrounding tissue that enables water to be drawn out of the filtrate. Which nephron structure is she describing? A. Proximal convoluted tubule. B. Collecting duct. C. Loop of Henle. D. Glomerular capsule. What do you think is the answer? Correct answer, CA, loop of Henle. Explanation: option C is correct because the loop of Henle uses a countercurrent multiplier system to create an osmotic gradient in the renal medulla, which allows urine to become progressively more concentrated as water is drawn out. Option A is incorrect because the proximal convoluted tubule performs bulk reabsorption of nutrients like glucose and amino acids, but does not create the countercurrent multiplier gradient responsible for concentrating the urine. Option B is incorrect because while the collecting duct is where final water reabsorption occurs under the influence of antidiuretic hormone, it relies on the concentration gradient already established by the loop of Henle. Option D is incorrect because the glomerular capsule surrounds the glomerulus and collects the initial filtrate produced by blood pressure-driven filtration, and it does not participate in concentrating the urine through osmotic gradients. The question describes a looping structure in the nephron that uses a countercurrent multiplier system to concentrate urine. That's the loop of Henle. The loop of Henle dips down from the cortex into the medulla of the kidney and then loops back up. As filtrate flows through the descending limb, Water leaves by osmosis because the surrounding medullary tissue is hypertonic. As filtrate moves up the ascending limb, salts like sodium and chloride are actively pumped out. But water can't follow because the ascending limb is impermeable to water. This creates an increasingly concentrated environment in the medulla. The deeper you go, the saltier it gets. That's the countercurrent multiplier. This gradient is what allows the collecting duct to later pull water out of the filtrate under ADH's direction. Without the loop of Henle building this gradient, the kidney couldn't produce concentrated urine at all. The proximal convoluted tubule does bulk nutrient reabsorption, not gradient building. The collecting duct benefits from the gradient, but doesn't create it. The glomerular capsule collects initial filtrate. No concentrating happens there. Here's a WGU D313 OA exam tip for you. Countercurrent multiplier, concentration gradient, and looping structure all point to the loop of Henle. Quick recall hook. Henle's loop creates the hypertonic medulla, H and H. The loop builds the salt gradient that makes concentrated urine possible. 46. A reproductive endocrinologist explains that sperm production occurs within specific tubular structures inside the testes. She adds that the testes are housed externally in a protective sac because developing sperm require a temperature slightly below the body's normal core temperature to develop properly. Which structure houses the testes, and what is its primary protective purpose? A. Scrotum protects sperm from excess heat. B. Prostate protects sperm from infection. C. Epididymis protects sperm from body temperature. D. Seminal vesicle protects sperm from acidity. Can you guess the answer? Correct answer. A. Scrotum protects sperm from excess heat. Explanation. Option A is correct because the scrotum is the external sac that houses the testes and protects developing sperm from the warmer internal body environment, since sperm require a temperature slightly below core body temperature. Option B is incorrect because the prostate gland is an accessory reproductive structure that contributes fluid to semen and can enlarge to constrict the urethra, but it does not house the testes or regulate their temperature. Option C is incorrect because the epididymis is a coiled tube where sperm mature and are stored after production in the seminiferous tubules, and it is not the external structure that maintains the cooler temperature for the testes. Option D is incorrect because seminal vesicles are glands that produce a fructose-rich fluid contributing to semen volume, and they do not house the testes or serve a temperature-regulating protective function for sperm development. Sperm production happens in the seminiferous tubules inside the testes, but the testes aren't tucked inside the body cavity. They hang outside in the scrotum. The reason is temperature. Sperm development requires a temperature about 2 to 3 degrees Celsius below normal core body temperature, which is around 37 degrees. If the testes were inside the body, that heat would damage developing sperm and reduce fertility. The scrotum solves this by housing the testes externally, away from the warmer core. The scrotum also has muscles that adjust how close or far the testes hang from the body, pulling them closer in cold conditions and letting them drop away in warm conditions, to maintain that ideal slightly below core temperature. The prostate gland contributes fluid to semen and can enlarge to constrict the urethra, but it doesn't house the testes. The epididymis is a coiled tube where sperm mature and get stored after production. It's inside the scrotum alongside the testes, but isn't the housing structure itself. Seminal vesicles produce fructose-rich fluid for semen. No connection to temperature regulation. Here's a WGU D313 OA exam tip for you. External sac, temperature protection, and housing the testes all point to the scrotum. Quick recall hook, scrotum keeps it cool. S for scrotum, C for cool. Sperm need to chill. 47. A medical student studies how testosterone production is regulated in the male reproductive system. He learns that a specific pituitary hormone binds to receptors on certain cells within the testes to directly stimulate testosterone release. Which hormone and target cell combination correctly describes this stimulatory pathway for testosterone production? A. FSH binding to Sertoli cells. B. Inhibin binding to anterior pituitary cells. C. Estrogen binding to hypothalamic cells. D. LH binding to Leydig cells. Can you guess the answer? Correct answer, D. LH binding to Leydig cells. Explanation. Option D is correct because luteinizing hormone binds to its receptors on Leydig cells in the testes and directly stimulates the release of testosterone, which is the primary hormonal cascade driving male testosterone production. Option A is incorrect because FSH binds to Sertoli cells to support spermatogenesis and sperm maturation, but this interaction stimulates sperm development rather than directly triggering testosterone release from the testes. Option B is incorrect because inhibin is a hormone secreted by the testes that sends negative feedback to the anterior pituitary to inhibit FSH release, and it is an output signal rather than a testosterone-stimulating input. Option C is incorrect because while estrogen does provide feedback to the hypothalamus in reproductive regulation, it is not the specific pituitary hormone that binds to testicular cells to directly stimulate testosterone production. Testosterone production in males is driven by a specific hormonal pathway, LH binding to Leydig cells. Luteinizing hormone is produced by the anterior pituitary gland and travels through the blood to the testes. Once there, LH binds to receptors on Leydig cells, also called interstitial cells, which sit between the seminiferous tubules. When LH locks onto those receptors, it directly stimulates the Leydig cells to synthesize and release testosterone. Testosterone then drives male secondary sexual characteristics, supports spermatogenesis, and maintains muscle and bone mass. FSH binding to Sertoli cells is a real pathway, but it supports sperm development and maturation, not testosterone production. Inhibin binding to the anterior pituitary is a negative feedback signal that slows FSH release. It's an output from the testes, not an input that triggers testosterone. Estrogen binding to hypothalamic cells is part of reproductive feedback regulation, but doesn't directly stimulate testosterone from the testes. Here's a WGU D313 OA exam tip for you. LH plus Leydig cells equals testosterone. That's the testosterone production pathway. Both start with L, that's your lock. Quick recall hook, L plus L equals T. LH plus Leydig equals testosterone. Two L's make a T. 48. A gynecologist explains the menstrual cycle to a patient and describes how one specific hormone is responsible for maintaining the thick, functional layer of the endometrium that supports embryo implantation during the luteal phase. If levels of this hormone drop, the endometrial lining breaks down and menstruation occurs. Which hormone maintains the endometrial lining during the luteal phase? A. Gastrin. B. Testosterone. C. Progesterone. D. Glucagon. Do you know the answer? Correct answer, C. Progesterone. Explanation. Option C is correct because progesterone is the key hormone of the luteal phase that maintains the thick stratum functionalis of the endometrium to support potential embryo implantation, and its decline triggers menstruation. Option A is incorrect because gastrin is a digestive hormone produced by G cells in the stomach that stimulates hydrochloric acid secretion and stomach contractions, and it plays no role in maintaining the endometrial lining. Option B is incorrect because testosterone is the primary male sex hormone produced by Leydig cells in the testes that drives male secondary sexual characteristics, and it is not responsible for maintaining the uterine endometrial lining. Option D is incorrect because glucagon is a metabolic hormone produced by alpha cells of the pancreas that mobilizes glucose from glycogen stores during fasting, and it has no function related to the menstrual cycle or endometrial maintenance. The menstrual cycle's luteal phase is dominated by one hormone, progesterone. After ovulation, the ruptured follicle transforms into the corpus luteum, which pumps out progesterone. Progesterone's main job during this phase is maintaining the thick stratum functionalis. That's the functional layer of the endometrium, the uterine lining where a fertilized egg would implant. Progesterone keeps that lining thick, vascular, and ready for a potential pregnancy. If no fertilization occurs, the corpus luteum degenerates, progesterone levels plummet, and without progesterone supporting it, the stratum functionalis breaks down and sheds. That shedding is menstruation, so the whole trigger for a period is the drop in progesterone. Gastrin is a stomach hormone for acid secretion, completely unrelated to the uterus. Testosterone is the primary male sex hormone from Leydig cells. Glucagon mobilizes glucose from glycogen stores during fasting. None of those maintain the endometrial lining. Here's a WGU D313 OA exam tip for you. Luteal phase, corpus luteum, endometrial maintenance, and menstruation when it drops, all of those signal progesterone. Quick recall hook. Progesterone supports gestation. It prepares the uterus for pregnancy. When it drops, the lining drops too. 49. A microbiologist explains that when pathogens breach the body's outer defense barriers, like skin and mucous membranes, the first internal immune cells to respond are those capable of engulfing and destroying the invaders through a process of cellular ingestion. Which type of immune cell represents this first line of internal defense against pathogens? A. Phagocytes. B. Antibodies. C. Helper T cells. D. Dendritic cells. What do you think is the answer? Correct answer, A. Phagocytes. Explanation. Option A is correct because phagocytes are the first line of internal immune defense when pathogens breach external barriers, and they engulf and destroy invaders through phagocytosis as part of the innate immune response. Option B is incorrect because antibodies are proteins produced by B cells during the adaptive immune response that mark pathogens for destruction, and they are not cells that physically engulf invaders as the first internal defense. Option C is incorrect because helper T cells are adaptive immune cells that regulate other immune cells by releasing cytokines, and they are part of the specific immune response that activates after the initial innate phagocytic response. Option D is incorrect because while dendritic cells do perform phagocytosis, their major specialized function is capturing antigens and presenting them to lymph nodes to initiate adaptive immunity rather than serving as the primary first responders. When pathogens break past the body's external barriers, skin and mucous membranes, the first internal immune cells to respond are phagocytes. Phagocytes are part of the innate immune system, which means they're non-specific. They don't need to identify a specific pathogen. They just recognize anything that looks foreign and eat it. The process is called phagocytosis. The phagocyte extends its cell membrane around the pathogen, engulfs it into a vesicle, and then destroys it with lysosomal enzymes inside the cell. Neutrophils are the most abundant phagocytes and arrive first. Macrophages come in next and continue the cleanup. This is all innate, immediate, and doesn't require prior exposure to the pathogen. Antibodies are proteins made by B cells during the adaptive immune response. They mark pathogens for destruction, but they're not cells and they don't engulf anything. Helper T cells regulate the adaptive response by releasing cytokines. They coordinate other immune cells but don't directly engulf invaders. Dendritic cells do some phagocytosis, but their primary function is antigen presentation, capturing antigens and delivering them to lymph nodes to activate adaptive immunity. Here's a WGU D313 OA exam tip for you. First internal defense, engulfing, and cellular ingestion all point to phagocytes. innate, non-specific, and immediate. Quick recall hook. Phago means eat. Phagocytes eat pathogens. They're the first to eat when invaders arrive. 50. A pediatrician explains to new parents that breastfeeding provides their newborn with a specific immune system component that offers temporary protection against infections without requiring the infant's own immune system to mount an active response. Which immune component found in breast milk provides this protective, passive immunity to the newborn? A. Helper T cells. B. Antibodies. C. Phagocytes. D. Cytokines. What do you think is the answer? Correct answer, B. Antibodies. Explanation: option B is correct because antibodies found in breast milk provide naturally acquired passive immunity to newborns, offering temporary protection against infections without requiring the infant's own immune system to actively produce them. Option A is incorrect because helper T cells are adaptive immune cells that regulate immune responses by releasing cytokines, and they are not the primary immune component transferred through breast milk for passive infant immunity. Option C is incorrect because while some immune cells may be present in breast milk, phagocytes are innate immune cells that engulf pathogens locally, and they are not the primary component providing the described passive immunity. Option D is incorrect because cytokines are signaling molecules released by immune cells to coordinate immune responses, and they are not the specific, protective immune component in breast milk that provides passive immunity to newborns. The last question wraps up with immunology, specifically passive immunity through breast milk. The immune component in breast milk that gives newborns temporary protection is antibodies. Here's the concept. A newborn's immune system is immature. It hasn't been exposed to many pathogens yet and hasn't had time to develop its own adaptive immune responses. Breast milk solves this problem by delivering the mother's antibodies, primarily IgA, directly to the infant. These antibodies coat the baby's mucous membranes in the gut and respiratory tract, providing a protective shield against pathogens the mother has already fought. This is called naturally acquired passive immunity. Naturally acquired because it happens through breastfeeding, not through injection. Passive because the baby's immune system didn't produce these antibodies. It received them ready-made from the mother. The protection is temporary because the antibodies eventually break down and the baby needs to build its own immune responses over time. Helper T cells are adaptive immune cells that coordinate other immune cells. They're not what gets transferred in milk. Phagocytes engulf pathogens locally, but aren't the primary immune component providing passive protection through breast milk. Cytokines are signaling molecules, not the protective molecules being transferred. Here's a WGU D313 OA exam tip for you. Breast milk, passive immunity, and newborn protection all point to antibodies. No active immune response from the baby means passive. Quick recall hook. Breast milk brings baby antibodies. 4 Bs. Mum's antibodies, baby's protection. You have reached the end of our first question set. For the free complete exam preparation, visit our website, link in the description, where you find thousands more practice questions and comprehensive study guides. Thanks for studying with us today. See you in the next video.